Hello, my dear students and future doctors. This is Vishwajit, your own zoology faculty.
So, dear students, the topics from this chapter we are going to cover, that is human excretory system, urine formation, mechanism of concentration of the filtrate, regulation of kidney function, the process of micturition, and lastly, the disorders of the excretory system. So, let's start without delaying. Okay. So, the first question we have to ask, that is, what is excretion? Obviously, we know that is one of the important process from life processes, along with the process of nutrition, digestion, circulation, right? And one of the most important process, that is excretion. So what is excretion basically? So in lower classes or in previous classes, you have studied that excretion is the process of elimination of metabolic waste from our body. See, inside our every cell, metabolic activities are going on. Metabolism is taking place. Now, what do you mean by metabolism? All the chemical reactions, all the chemical reactions. And during the time of this chemical reaction, we'll get few components which are green signal, are good for our body, and obviously we'll be producing something inside our cells which are not at all good for us. Now, if you keep those waste products inside those cells, gradually they're gonna kill us. So that's why here comes the necessity to eliminate them out, from the body, and primarily that is called excretion. Okay, so let's see what NCERT is saying to us. So the NCERT, according to NCERT, animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chloride, phosphate, sulfate, etc., either by metabolic activities, we know obviously, and the second thing is, or by other means like excess ingestion. So if you are drinking excessive amount of water, if you are taking excess salt in your diet, so that gonna also act as excreta or waste product of your body. Now here we can see metabolic activities is written here. Now, how it's related to our topic? So, let's take one of the metabolic activities, the reaction of respiration, right? So, in respiration, what happens? We have glucose inside our cells, C6H12O6, and that is getting oxidized by oxygen. And after oxidation, what is formed? Carbon dioxide, water, and energy in the form of ATP is formed. Now these products, our body will treat these products as excreta. So these are metabolic waste. You need to eliminate them out from the body. Okay. Now here we can see that our body accumulates different types of excretory products. We will be categorizing them. One category that is of nitrogenous waste, another category that is of non-nitrogenous waste. So let's talk about nitrogenous waste. Ammonia, urea, and uric acid. They are the major nitrogenous waste produced inside our body. And when I'm talking about non-nitrogenous wastes, they are this carbon dioxide, water, ions like sodium, potassium, ions like chloride, bicarbonate, even the proton, H plus ion, a major excreta of our body, along with phosphate ion, PO4 3 minus, and sulfate ions, SO4 2 minus and PO4 3 minus. So this will come under the category of non-nitrogenous waste, and ammonia, urea, uric acid, they will fall under nitrogenous waste. Okay. Now let's talk a little bit more about this ammonia, urea, and uric acid. So when I'm talking about ammonia, this is NH3. Now our body will be converting this ammonia into ammonium, NH4 plus, and that will be easily diffused out with the help of water. This is ammonium. Now, which animals will be eliminating out or excreting out their metabolic waste in the form of ammonia or ammonium? Gradually, we're going to learn them. Okay. Now, urea. Urea, the second nitrogenous metabolic waste produced by many organisms. So the structure of urea will be like this. CO NH2 whole 2, the formula of urea. Now the third one is uric acid, and you need to know this. This is the derivative of purine base. Right? So somehow the structure will be like this. So this is the ring structure of purine, and with this you will be finding nitrogen and carbon inside the rings. So let's move forward. Now it said that these substances have to be removed totally or partially. When we are talking about all these excretory products, few products will be removed totally from our body. Now which product you need to be removed totally? Ammonia, NH3, as because it's very toxic for us. And if someone asks you, tell me about excretory product which you need to retain in a particular concentration in our body, so you'll be removing them partially. So the answer will be urea. Right? So the urea is retained by our body to maintain the osmolarity, or that helps in the osmoregulation to maintain the tonicity of the fluid of our kidney. Right? So when I am talking about urea, our kidney retains certain amount to regulate or to maintain, better to be said, to maintain osmolarity or tonicity of fluid inside kidney. So we'll be elaborating this. Let me give a brief explanation of this particular thing. So basically, the presence of urea is reabsorbed through distal part of collecting duct of our nephron. And what is nephron? The structural and functional unit of our kidneys. So urea is getting reabsorbed. Now, why our body is reabsorbing urea? Though a certain amount of urea is getting excreted, but a certain amount of urea is getting reabsorbed, that is responsible for maintaining the hypertonicity of the medullary region of our kidney. So if this is kidney of us, outer region is called cortex, inner region is called medulla, and the nephrons are reabsorbing urea to increase the osmolarity or tonicity of the medullary region of kidney. So that is making the fluid or interstitial fluid present into the medullary part, medullary tissue of the kidney, and maintaining the hyperosmolarity or hypertonicity. Okay. Now, basically, we'll be emphasizing on discussion of nitrogenous waste, that is ammonia, urea, and uric acid in this chapter. Ammonia, urea, uric acid, the three major excretory waste as well as nitrogenous waste. We will be differentiating. When I'm talking about ammonia, first we'll compare their toxicity. So, it's highly toxic. Ammonia is highly toxic. For urea, the toxicity is moderate. And the least toxic is uric acid. So if you want to arrange the rank of toxicity, first it will be ammonia, then it will be urea, then it will be uric acid. It's like that. Now, if an animal is excreting them out, what medium it's using? So you can expect that they will be dissolving the excreta inside the water and they will be eliminating out from through their body, maybe through a particular organ, that is kidney, or maybe through gill surface, or maybe simply through the body surface. Now, ammonia, if you want to dissolve ammonia, you need a lot of water. So water requirement is quite high. Water requirement, it quite high for ammonia. Now if you ask me, sir, how much water is needed to dissolve 1 gram of ammonia? You need near about 500 to 700 ml of water, right? So to dissolve 1 gram ammonia, you need near about 500 ml of water. Now if we talk about urea, water requirement is moderate. Again, some medium, and it needs near about to dissolve 1 gram, you need to use 50 ml of water. Now for uric acid, for uric acid, the water requirement is very low or the lowest, and you will be needing to dissolve 1 gram of uric acid. You need only 5 to 10 ml of water. So you can guess the solubility of them. So solubility of ammonia is quite higher or the highest among them, very much soluble in water. Solubility of urea is again moderate. This is also very much soluble in water. But the solubility of uric acid is very low, because uric acid, they will form solid crystals. Okay. Now ammonia can be excreted directly as the ammonia gaseous form dissolved or diffused into the water, or it may get converted into ammonium NH4 plus, dissolved or diffused out, diffuses out through gill or body surface. Got it? Urea is excreted as such, as liquid form with urine. So let's come to uric acid. Uric acid excreted as pellet or solid crystals or paste with feces. So basically, the animals who are excreting out uric acid, they produce uric acid in the form of pellet, paste or solid crystals, and they excrete it out or they mix it with the alimentary canal while they are passing out their fecal matter or stool. So with the feces or their stool, they will be eliminating out the uric acid. Now the animals who exhibit excretion of ammonia, we call them ammonotelic animals. The animals who exhibit excretion of urea, we call them ureotelic animal. The mechanism is called ureotelism. The animals who excrete out uric acid, we call them uricotelic animal. The mechanism is called uricotelism. Now, as because the ammonia or ammonium is excreted out or eliminated out with the help of their gill surface or the skin surface, role of kidney, there is no such important role involved for kidney. So role of kidney, there is no role of kidney, but it's the kidney will be filtering out the urea from the blood and will be producing the urine, and then it will be urinating. So the role of kidney is quite important. So role of kidney is involved for the excretion of urea. But when you are talking about uric acid, again there is no role of their kidneys specifically, is because it is released with the help of their fecal matter through their alimentary passage, right? So here also for excretion of uric acid, the role of kidney is limited. Now let's talk about the mode of adaptation means, if you are asked that the animal kingdom is highly diversified, now which animals exactly they tend to excrete ammonia, which animals will be excreting urea, and which all animal will be excreting out uric acid. For that, we need to conclude this with the story of evolution. See, in the beginning, there was water bodies, right? So the animals who are living into water, the aquatic adaptation, the aquatic mode of adaptation, like freshwater fishes, tadpole, larva, aquatic arthropods, they have a lot of water to use. So that's why with the help of their gill or body surface, directly they release ammonia or ammonium. So when you talk about aquatic adaptation, then the animals will become ammonotelic. Now we'll be excreting out urea. Few aquatic animals like sharks and all, or the marine fishes, right? And a few land animals like us, mammals, we are also ureotelic, and few amphibians. So when you're talking about mode of adaptation, it may be aquatic, it may be terrestrial. Depending upon the situation, okay, depending upon their habitat, depending upon the external environment, depending upon the medium in which they are living in, and the tonicity of the medium, right? And when you're talking about uric acid, now this is very much crucial. Now gradually the fish, they will come out and they will become amphibians like frogs. Now these frogs want to inhabit it into the land zone, right? We call them amphibia. So gradually the environmental was drying day by day, and the water bodies were shrinking. Water bodies were shrinking. So there is more population in the water. They are competing with each other. A few of the fishes, they were trying to come out of the aquatic body, and that given rise, or that evolved the formation of amphibians, and the dryness or the drought was increasing day by day. The sun was getting stronger. There was no rainfall, though initially abundance of rainfall was there. There was enough moisture in the environment, enough water bodies were present. But as because the weather was becoming drier and hotter, gradually there will be a modification into these amphibians, and they will become reptiles. When they will become reptile, the environment is quite dry, and the zone is terrestrial zone, land. So the animals who are inhabiting or living into the land, and they are facing dry or drought kind of condition, so they cannot afford to lose water for the process of excretion. So that's why nature switched their mode of excretion from ammonotelism or ureotelism to uricotelism, as because excretion of uric acid requires only a very little amount of water. 1 gram of uric acid requires only 5 or even less amount of water to be excreted out. So when you're talking about the mode of adaptation, uricotelism is exhibited by the terrestrial animal. Terrestrial adaptation specifically. Okay, now let's learn about the examples which animals are ammonotelic. Porifera, Nidaria, they are monotelic. Freshwater fishes, mostly they are bony fishes, freshwater fishes and bony fishes, few brackish water and also few marine water bony fishes are also a monotelic. Okay, then aquatic amphibians, they also excrete ammonia. Tadpole larva, they're also aquatic. Tadpole larva amphibia, they're a monotelic, right? And obviously aquatic arthropods, like shrimp, prawns, daphnia, crabs, they also will be excreting out ammonia. Now let's talk about rheotelic animals. Mammals, we are rheotelic. Then amphibians, frogs, toads, they'll also pass urine like us, right? Marine cartilaginous fishes, are also will be excreting out urea. Okay, now let's come to the uric acid excretion part. Reptiles, birds, terrestrial insects, land arthropods, and few land snails, they also exhibit uricotelism. So again, once a rapid recap of ammonotelism, ureotelism and uricotelism. Ammonia is the most toxic nitrogenous waste, whereas uric acid is the least toxic nitrogenous waste. I'm repeating it again and again is because there are very much possibilities, high possibilities are there that question will come from this particular topic of excretion. Yes, and the animal either they will become monotelic or ureotelic or uricotelic, that will be chosen by the nature in the course of their evolution, right? Ammonia is excreted with the help of their gill surface or the body surface of fishes as ammonia, directly as ammonia, or most easily in the form of ammonium ions. Urea in the form of urine excreted out, and uric acid in the form of pellet, in the form of pellets or paste with stool or fecal matter, right? So let's move to the next topic. Okay, so when we're talking about excretion, we need to talk about osmoregulation. They are interconnected, right? Because just now we have studied the different animals and their mode of excretion, and after this we are going to study about different structures related to excretion or excretory structure, excretory organs. Now most of the times we'll observe that the evolution or the development of excretory structure inside animal body was primarily due to maintain the water and the electrolyte or ionic balance inside the body. That is called osmoregulation, and also they are helping in the process of elimination of nitrogenous waste. Right? So we know the excretion means elimination of metabolic waste. So removal of waste by the mode which are produced by the mode of metabolism. They can be metabolic waste, or they can be accumulated into our body by the method of or after excess ingestion, excess eating, excess drinking. And what is osmoregulation? Electrolyte and water balance inside our body. Electrolyte and water balance inside our body. You know this particular terminologies, right? Isotonic solution, hypotonic solution, hypertonic solution. Now what is this isotonic, hypotonic and hypertonic? For if we talk about human being, for us, the tonicity of our blood or the body fluid, blood, lymph or other body fluid, near about 300 milliosmol per liter. That is the osmolarity or tonicity of our body fluid. Now, whatever phenomena will be happening inside our body, we'll try to manage. We know about homeostasis. Our body has a capability to maintain a range of everything, right? So, our body will try to manage or maintain the osmolarity of everything which are happening inside our body, inside the cells, and inside the intercellular space or inside the tissue space, to maintain that tonicity equal to 300 milliosmol. That will be isotonic. If something in compared to our body fluid having the similar kind of concentration into it, so we will call that solution as isotonic solution. If the osmolarity or tonicity is less into this, we will be calling that solution as hypotonic solution. And if the osmolarity in compared to our body fluid or blood is higher, there are more electrolytes, more ions, more solutes dissolved into the solvent, then we will be calling this as hypertonic. Like for an example, we have taken 3 glasses with water. What's that? Okay, so we'll take 3 glass of water of similar volume. In the first glass, I will add salt. Suppose I will add only half teaspoon of salt. The second glass, I will add 2 teaspoon of salts, and in the third glass, I will add 4 teaspoon of salts. Now, when you will be drinking the water, so what you will be feeling? Which one will be the hypertonic solution? Which one will be the hypertonic solution? The glass, 3 glasses, glass 1, glass 2, glass 3. Suppose I have kept 500 ml of water inside them. First glass, half teaspoon of salt. Second glass, 2 teaspoon of salts. Third glass, 4 teaspoon of salt. So third one will be the hypertonic in compared to glass 1 and glass 2. Glass 1 will be the hypotonic in compared to glass 2 and glass 3, right? But suppose I have taken another glass and I will add again 2 teaspoon of salts, so that will be isotonic with glass 2, right? So every time our body is doing metabolic activities inside that, there will be electrolytic imbalance, but our prompt reaction, the homeostatic mechanism of our body will rectify that. And for that, our excretory structures will help us to do so. Okay, now here an account of evolution of different excretory structures are given here. So basically, in lower invertebrates, you won't be finding any specified respiratory structure. They will be excreting out their excreta with the help of their body surface simply, right? But gradually, as because there is advancement or evolution, you can observe first protonephridia in some of the animals. Then the modified version of protonephridia will be nephridia. Protonephridia is the old version and the advanced version is nephridia. You can also call it metanephridia. Metanephridia, meta means advanced, developed, and the developed version of nephridia will be kidney. And obviously when you study kidney, there are different types of kidneys are there. Like if you talk about the evolution of kidney, first you can observe protonephric kidney or protonephros kidney, the primitive kidneys. Advancement of protonephric kidney, you can find mesonephric kidney. In some animals you may get opisthonephric kidney. In higher classes you are going to learn them. So protonephric kidney is the old kind of kidney. Advancement of protonephric kidney, you will find mesonephric kidney. As advancement of mesonephric kidney, will get the advanced or the modern type of kidney that is called metanephric kidney. Now do not confuse between metanephric kidney and metanephridia. So the evolution of excretory organs are like this, from protonephridia to nephridia or metanephridia, from nephridia to kidney. In the beginning, the type of kidney you can observe, there is protonephric kidney. On evolution, we will get mesonephric kidney, and the highest level of development or advancement is seen in metanephric kidney. Let's move forward to excretory structure in different animals in animal kingdom. Okay. So when you talk about simple invertebrates, primitive invertebrates, now if you talk about Podifera, Cnidaria, Ctenophora, they don't really have specifically Podifera and Cnidaria, right? As sponges, hydra, jellyfish, they don't have excretory structure. The first time excretory opening or excretory structure you will be finding into the body of Ctenophora, then later on in platyhelminths. Okay, so basically they have a simple tubular structure, and upon advancement of simple invertebrates we will be getting complex vertebrates, and in the body of inside the body of complex vertebrates we will be getting complex tubular structure. Obviously kidney can be termed as complex tubular structure. Our one kidney is made up of 10 lakh or 1 million of complex tubular nephrons. You know it, right? Okay. Now let's start from the beginning of the animal kingdom. Platyhelminths, flatworm, rotifers. Now this particular phylum, we call it rotifera, is not given in NCERT. Now as per our NCERT knowledge, we can somehow consider these rotifers within the phylum Aschelminths, right? Okay. So platyhelminths, rotifer, though the rotifers are categorized into a different phylum nowadays, that is phylum Rotifera. Few annelids or initial annelids, earthworm, leech, nereis, they are quite advanced in annelida. They are quite advanced annelida. But here they are talking about the old or the initial annelids. And we got one higher order animal as well, that is cephalochordate. When someone asks you cephalochordate, you need to give example of Amphioxus. Cephalochordate, first successful chordate animal. Okay. So all these animals, flatworms, rotifers, primitive annelids, cephalochordates like Amphioxus, they have protonephridia. They have protonephridia. The protonephridia can also be said as phlegm cells. The protonephridia also can be said as solenocyte. Now, why it's so? Because of their special structure. So this is the solenocyte cell or the phlegm cells. Now here you can get a tuft of ciliary structure that resembles like the flame. That's why we call them phlegm cell or solenocyte. Now this is tubular cell to pass out the excreta. Now let's come to the next group of animal, annelids or developed annelids. They have metanephridia or nephridia. When you talk about insects, these all falls under arthropods. Now arthropod is the largest phylum in animal kingdom and they are highly diversified. Insects, mostly they are terrestrial and they have Malpighian tubules. Insects have Malpighian tubules. Do not confuse with Malpighian body, right? Malpighian tubules are excretory structure of insects, but Malpighian body, they are excretory structure, associated excretory structure or present within our nephron, Malpighian body. So Malpighian body present inside the nephron, but Malpighian tubules, excretory structure, individual excretory structure of insects. When you have studied cockroach, you might have studied about these Malpighian tubules into the junction of their midgut and hindgut. The junction of if you studied the alimentary canal of cockroach, excretory system of cockroach, there you will be getting the junction of midgut and hindgut. There you will find 100 to 150 yellow colored thread like fibrous structures. They are collecting excreta from their hemolymph or colorless blood and releasing the uric acid into the hindgut. They are basically Malpighian tubules. Okay. Now, when you're talking about aquatic arthropods, now who are aquatic arthropods? Crustaceans. Who are crustaceans? Prawn, shrimp, mantis, water mantis, daphnia. They are aquatic arthropods. So they have green glands for excretion, and these are also called as antennal gland. Green glands or antennal glands. Now let's come to another group of arthropods. We call them arachnids. Now who are insects, who are crustaceans, and who are arachnids? Insects are those arthropods who have 3 pairs of legs and 1 pair of antenna. Crustaceans, mostly they have 5 pairs of legs and 2 pairs of antennas, and mostly they are aquatic in nature. Now, if you talk about arachnids, examples are written here. Spiders and scorpions, they have 4 pairs of legs or 8 legs. You might have seen spider in your home, right? Now, they have coxal gland. They have coxal gland as their excretory structure. And when you're talking about vertebrates, for most of the vertebrates, the excretory organ is kidney. Obviously, in few there will be protonephric kidney, in few there will be mesonephric kidney, in few there will be metanephric kidney. Now we got another group with dual mode of excretion. Now what's that? Dual mode of excretion. Now, if we talk about earthworm, a very good example will be performing dual mode of excretion. In dry season, the earthworm will become ureotelic, will be excreting urea, right? But in monsoon, when there is abundancy of moisture and water, it start excreting ammonia. So that is called dual mode of excretion. Crocodile, sometime it will become ammonotelic, sometime it will become uricotelic. So dual mode of excretion, let's make a different slide of it. Dual mode of excretion is exhibited by earthworms, crocodile, ascaris. Like if I'm talking about earthworms, in monsoon, they become ammonotelic. They excrete out ammonia. In dry summer, they release urea. That's why we call them friends of farmer. They become ureotelic. Crocodiles, crocs, sometimes they become ammonotelic, sometimes they will become uricotelic. And similarly, ascaris, they are also ammonoureotelic. Means sometimes majorly they will be excreting out urea, but sometimes they also can excrete out ammonia. So that was all. Now we'll be moving forward to the next topic, that is human excretory system. So first, the NCERT says that human excretory system is made by one pair of kidneys, one pair of ureters, one single urinary bladder, and one single urethra. So if someone asks you the accountability of the organs related to the excretory system of human being, so here they are. So here you can observe two kidneys are there, and their apparent situation, their apparent position, you can observe that they are not in a same level. So this particular kidney, this is the left kidney, and this particular kidney, this is the right kidney. Now, why the position of right and left kidney is not even? Due to the positioning of liver, right, into our abdominal cavity. The whole system present into our lower abdominal cavity towards the pelvic cavity, and the position will be towards the dorsal wall, towards the dorsal lower abdominal or pelvic wall. Precisely we will obviously learn, but first let's mark them properly. So you got two kidneys, left kidney and right kidney, and the superior pole of kidney we can get these are adrenal gland, though they are not associated with human excretory system. From kidney carrying urine, these ureters are coming down. So these two are the ureters, left ureter and right ureter, and the ureters will be releasing the urine into urinary bladder. Now we have blood vessels. Obviously, when we are studying about the function of kidney, we need to talk about the blood supply. So this is the branch of aorta. This is called dorsal aorta, which is supplying oxygenated blood into the kidney, as well as this will be supplying the nitrogenous waste, and inside the kidney the nitrogenous waste will be getting filtered out, and through renal veins they will be passed into the vena cava, inferior vena cava. So the major blood vessels you can observe here, this one is a dorsal aorta, a branch of aortic arch, and this one is inferior vena cava. And from dorsal aorta, the branches of the blood vessels, they are going inside the kidney. They are called as renal arteries. This is renal artery, and this will be renal vein. The vein which is coming out from kidney and entering and merging with inferior vena cava, they are renal veins. So that was all about the superficial, the structure of human excretory system. I'm extremely sorry. The superficial structure of human excretory system. Okay, one more thing, I need to mark it here. So this particular pipe coming out from the urinary bladder, that is called urethra. At the end of this urethra, you are gonna get the urethral opening or urethral meatus. Now here I need to mention that the urethra of male is quite longer, 15 to 20 centimeter long, but in case of female, urethra is quite shorter, just 3 to 5 centimeter. So I've already learned the human excretory system, the main parts, that is one pair of kidneys, one pair of ureters, one single urinary bladder, and urethra. Okay. Now the question can be asked conceptually that why the apparent position of both the kidneys, left and right kidneys, are not equal. They are uneven in nature in terms of their distance and their position. Okay. So it's because of the positioning of liver into our abdominal cavity. So you know that shape of the liver is kind of a triangle. So that's why the right side of the liver is little bit downwards in compared to the left side of the liver, and that pushed the right kidney downwards and left kidney little bit upwards. So that was the reason behind the positioning of both the kidneys. Now we'll talk about the kidney. Basically we'll be studying about the morphological aspect. Now what do you mean by morphology? Now if you are studying a kidney from outside, you have to check what is the length of that particular kidney, what is the width, what is the thickness, what is the color, what kind of size, what kind of shape it has. So let's talk about the morphology of kidney. So before that, I want to give up information about kidney, that our kidney, human kidney, that is particularly metanephric type. Now already we are knowing that kidneys are of mainly three types: protonephric, mesonephric, and metanephric. Now, metanephric is a highly advanced or highly developed kind of kidney. Okay, so that's why the type of our kidney is metanephric type. Now, second question will be asked, what is the location of kidney in our body? So, the location of our kidney is present in dorsal inner wall of abdominal cavity. Present into the dorsal inner wall of our abdominal cavity in between. Now, this is the precise location in between T12 and L3. Now, what do you mean by this? T12 means the last thoracic vertebrae, L3 means third lumbar vertebral. So, you know about the vertebral bone, right, in our body. So, if this is our body, suppose it's a lateral view. So, from here you can observe vertebral bones towards the dorsal side. Now, this is the dorsal side of our body and this is the ventral side of the body. This is the ventral side and this is the back side is the dorsal side. So, vertebral columns are located dorsally, obviously. And the vertebral bones are like this. They're cervical, thoracic, lumbar, sacral and coccygeal. These 5 kind of vertebral bones are there. There are 7 cervical vertebral bones are present. 12 thoracic vertebral bones are there. 5 lumbar vertebral bones are there, and 1 fused sacral and 1 fused coccygeal bone you'll be finding into human body. Now, somewhere if I'm suppose naming this particular bone as T12, that means this is the last thoracic vertebral bone or last thoracic vertebrae. And if I'm marking this particular bone as L3, so that means this is the third lumbar vertebral bone or third lumbar vertebrae. So, in between these two bones, you'll be finding the positioning of our kidney towards the dorsal wall, of our body. This is towards dorsal. Kidneys are not present towards the ventral wall. Suppose if this is the body cavity, this is the peritoneal wall or the peritoneum, the cavity, the wall of our body cavity is called peritoneum. Now, the kidney is present outwards to the peritoneal cavity. That's why you can also call kidney as a retroperitoneal organ. Yes, so that's also written here. Kidney is said to be one of the retroperitoneal organ. Why it's retroperitoneal organ? This is the outer wall of our body, and this is this inner wall or peritoneal lining. Lining wall of our body cavity is called peritoneum or peritoneal lining, and the cavity inside will be called peritoneal cavity. Now, these two are kidneys. Now, you can observe that these kidneys are present outwards to the peritoneal cavity. Now, this kind of cavities are called peritoneal cavity. This kind of cavities are called peritoneal cavities. And if you will find like if some organs you are observing present inside the peritoneal cavity, you can call them as visceral organ. You can call them as visceral organ. And if organs are present outwards to this peritoneal lining, you can call these organ as retroperitoneal organ. So kidneys are also retroperitoneal organs. Ovaries are also examples of retroperitoneal organs. Few parts of your alimentary canal is also falls under this retroperitoneal organ. So kidneys are also retroperitoneal because of their special position that is outwards to the peritoneal lining or the lining of our body cavity, which is not completely covered by the peritoneal wall. There is only one-sided covering. There will be finding only one-sided covering of peritoneal wall, and another side is free of peritoneal lining. That's why we are uttering the word outwards to the peritoneal cavity. Okay. Now let's talk about the color of our kidney. The color will be reddish brown or red brown in color. If we talk about the shape of our kidney, we can see here just like kidney bean or bean shape. Now let's talk about the dimension or the size of our kidney. So when you're talking about the size, we have to talk about length, width and thickness. So if you're talking about length of kidney, it's 10 to 12 centimeter long. If I'm talking about the width of kidney, this is 5 to 7 centimeter long. And if you're asking about the thickness of our kidney, if you're asking about the thickness, so this is the thickness of our kidney, then it will be 2 to 3 centimeter only. Now also, if you study NCERT, the weight of kidney is also given there. And the weight of kidney in between 120 to 170 grams. So you can say at an average 150 gram. Now kidney is covered by a special kind of fibrous and epithelial tissue, and that covering is called renal capsule. So the outer tough covering of kidneys are called renal capsule. Now there are three kinds of capsule you will be finding. One is fibrous capsule. The immediate covering of kidney, that is called fibrous capsule. After that you will be finding adipose capsule made up of fatty tissue, and then you will be finding a special kind of fascia called renal fascia. So it's like this somehow. If this is the kidney, the first covering we are going to get, that is the fibrous capsule. After that we are going to get the adipose capsule. After that you are going to get the renal fascia, right? Now this part, this concave part is facing the peritoneal cavity. Okay, and this is towards the outwards to the kidney, which has the convex lining, and this is called the superior pole of kidney or anterior pole of kidney. This is called the inferior pole of kidney. Towards the superior pole, you will be getting what? You will be getting adrenal glands. Okay, so these are the things we are talking about. So specifically, when you're talking about renal capsule, it's made up of mainly made up of tough fibrous tissue. The covering of kidneys are called renal capsule, mainly made up of tough fibrous connective tissue, right? Tough fibrous connective tissue. Then we'll be getting adipose tissue and renal fascia towards the anterior part of the kidney. Okay, now let's go into the anatomy and in detail explanation of our kidney. So as I was saying that this portion is called the outer convex plane of kidney. Kidney also has inner concave plane, and towards this inner concave plane you are going to get the opening through which the blood vessels, means arteries, veins and nerves will enter into the kidney, and that precise opening is called as hilum or hilum, right? So what is hilum or hilum of kidney? Towards the inner concave plane there is an opening through which blood vessels like renal arteries, renal veins will come in or go out, and nerves enters or exits into the kidney or outside the kidney. That opening is called hilum, or that concave portion is called hilum. Okay. Now let's detallify the inner structure of kidney. First, we have to denote the outer capsule. Then, majorly, kidney has outer part and inner part. So, this outer part of kidney is called cortex or renal cortex, and the inner part of kidney is called as renal medulla. And into the renal medulla, you'll be finding this kind of pyramidal structure made up of renal tubules, collecting duct, or loop of Henle. This kind of parts of a nephron will form this pyramid kind of structure into the renal medullary region, and we call that part as medullary pyramid. So when we are talking about medullary pyramids, in a single kidney, at an average, 8 to 12 medullary pyramids can be found. Now, if you are asked that in human body, means in a body there will be two kidneys. So, if you consider both the kidneys, there will be you will be finding 16 to 24 medullary pyramids. But for a single one, 8 to 12. Okay. Now, if you check the structure of medullary pyramid, the pointed conic edge part of every medullary pyramid, that is called renal papilla. Renal papilla. So that's also part of medullary pyramid. The pointed conic edge of every medullary pyramid is called renal papilla, and together all this region is called renal papillae. Okay. Now, one very important thing we can observe here, that if this is the tissue of cortex, right? So the cortex, the tissue of cortex will enter into the medullary zone. The tissue of cortex will enter into the medullary zone in between the medullary pyramids. So that's forming a column kind of structure, and we call that renal columns or columns of Bartni. one column is column of Bartni or renal. You can also call it as renal column. Columns of Bartni can be also called as renal column. Now, how you can define renal column? This is the cortex, the columns of cortex in between the medullary pyramids. The columns of cortex into the medulla, present in between medullary pyramids. It's called renal column or columns of Bartni. Okay, now let's move forward. Here you can observe the nephrons as well. Nephrons majorly found into the cortical region. You can call them cortical nephrons, and the nephrons present near to the medulla, and their parts like Henle's loop deeply embedded or penetrated deep down into the inner medullary zone. They are called juxtamedullary nephrons. We'll be detailing about them. So, present on the basis of their location upon like on kidney, the nephrons which are present towards the cortex and hardly getting penetrated into the medullary pyramid region, they are called cortical nephrons. And the nephrons which are present near to the medulla and their Henle's loops are deeply penetrated into the medullary pyramids, they are called juxtamedullary nephrons. Yes. Now, this V-shaped part, a funnel kind of structure which is coming out of a single kidney, that is called renal pelvis or pelvic region. It is called renal pelvis or pelvic region of kidney, the funnel kind of structure. The funnel kind of structure. Okay. Now, when the renal pelvis is getting inside each kidney, it will form few finger-like structure, and those fingers are called major calyx. And further, these finger-like structures, they are divided into numerous short fingers kind of structure, and we call them as minor calyx. So, minor calyx, if we are going from inside towards the outside, inside you will be getting minor calyx, then minor calyx joins together to form major calyx, and all the major calyx will join together to form renal pelvis, and the renal pelvis will come out as a pipe, and this pipe is called as ureter. So, the part after the renal pelvis, that is called ureter. So, it's like this somehow. This is the pelvis region. The pelvis is divided into major calyx divided into minor calyx, and the pelvis will be continuing as ureter and join with the urinary bladder. So, let us mark them. So, this is ureter, this is pelvis, this is major calyx, these are minor calyx. And these minor calyx, they are connected to renal pyramids. The number of renal pyramids somehow equal to the number of minor calyx. So how many renal pyramids are present into a kidney? 8 to 12 or 13, right? So that's why it's written here in a kidney, the number of major calyx, they're equal to the minor calyx, they're equal to the renal pyramid. Now how many renal pyramids are there? 8 to 12 renal pyramids are there. When we'll be talking about the number of major calyx in a particular kidney, then the number will be 4 to 5, half of the number of minor calyx, right? So this is how your kidney will be made. Okay, now let's talk about the plumbing system of kidney. Not plumbing system, blood vascular system of kidney, how blood vessels are going inside and coming out. So this is the renal artery bringing oxygenated blood as well as the blood with nitrogenous waste inside the kidney for the process of filtration. So the blood vessel which is responsible for taking oxygenated blood as well as impure blood in terms of presence of nitrogenous waste is called renal artery. It's called renal artery. And the other segment, the another blood vessels which is bringing out the deoxygenated blood as well as pure blood in terms of the vein which is bringing out the blood from the kidney after filtration, that is free of nitrogenous waste, and this is called renal vein. This is called as renal vein. So the blood vessel which is uh, taking in blood inside the kidney for the process of filtration, that is renal artery, and the blood vessel which is taking out blood from the kidney after the process of filtration, that is renal vein. So here tricky question can be asked that if you fetch some blood from renal artery and inject out some blood from renal vein, so what's our mindset? We think that the blood present into the artery, they are pure, and the blood present into the renal vein, they are impure in terms of presence of oxygen and carbon dioxide. So in terms of oxygenated and deoxygenated blood, the arterial blood is oxygenated and the venous blood is deoxygenated. But if we consider the presence of nitrogenous waste or presence of excreta, then which will be pure one? Then the blood present into the renal vein, that will be pure one in compared to the blood present into the renal artery, because the renal artery that carrying blood, that is rich in nitrogenous waste, urea, ammonia, as well as other excreta. Okay, now the next thing we have to study the path of urine. So inside the nephron, 10 lakhs of functional inside the kidney, 10 lakhs of functional nephrons are present. Then their function is to filter the blood and to eliminate out the nitrogenous waste and to mix it with water to form urine. Now urine is getting prepared inside the kidneys, particularly inside the nephrons, and when the urine will be coming out, when the urine will be coming out from kidney, then what kind of pathway they're gonna follow? Okay, so here you have to study the pathway of urine. So let's start with collecting duct, that is a part of nephron. Obviously the detailed structure of nephron we're gonna study. So inside the collecting duct, real urine is prepared. Then collecting duct will open into a wider narrow channel, that is called duct of Bellini. Collecting duct will be opened into a narrow drainage system, it is called duct of Bellini. Now the duct of Bellini will be leaving renal pyramid or medullary pyramid. Now after that it will enter into the minor calyx. From minor calyx the urine will enter into the major calyx. From major calyx the urine enters into the renal pelvic region, this region, and after that the urine will be thrown into the ureter, and obviously from ureter, at the end of ureter, you'll be finding urinary, at the end of ureter, you'll be finding urinary bladder, and ultimately from urinary bladder, the urine will be released out through urethra. Through urethra, we'll get an opening or urethral meatus. After urethra, we'll get opening called urethral meatus. Okay. Now, you know that urethra is long in case of males and short in case of female. The urethral meatus or urethral opening, is separate in case of female, and urethral meatus will be associated or will be opened through the penis or the external genitalia in case of males. You know already. Okay. Now it's time to moving forward towards the structural and functional unit of kidney, that is nephron. So what is nephron? Structural and functional unit of kidney. Right. Now next thing, how many nephrons are present in a single kidney? The answer will be 1 million per kidney. 1 million nephron per kidney. That means 10 lakhs nephrons in a kidney. Now if you are asked that how many nephrons are present in human body, then multiplied by 2 millions of nephrons present in human body, because if one kidney has 10 lakhs or 1 million, then two kidney will have 2 millions or 20 lakhs, as simple as that, right? Okay. Now let's talk about the parts of a nephron. So the parts of nephron are divided into glomerulus, first part of nephron, a tuft of blood capillaries, and then renal tubules, the tubular part of nephron, right? So what is glomerulus? Glomerulus is tuft of capillaries, tuft of blood capillaries, bunch of, tuft means bunch of blood capillaries, you can call it glomerulus. Now when you talk about renal tubules, what are the parts of renal tubules? The first part of renal tubule will be Bowman's capsule. What is Bowman capsule? It is a double-walled cup. There is a double-walled cup-like structure. It is the outer wall. It has one inner wall. So the double-walled initial part of renal tubule is called Bowman's capsule. After that, we'll get PCT, proximal convoluted tubule. Proximal convoluted tubule. Now, what is the meaning of this proximal convoluted? Proximal means which is present near. So this proximal convoluted, proximal means present near to the glomerulus. Convoluted means coiled. Coiled, it's a coiled structure, and tubule means small pipelines, small tubes are called tubules. Now, LOH or HL in NCERT it's mentioned as HL, that is loop of Henle or Henle's loop. Loop of Henle or Henle's loop. Now DCT, the full form after loop of Henle will get DCT, that is distal convoluted tubule. Distal convoluted tubule. Now meaning of the distal, distal means far, the distance. Proximal means proximity, near. Convoluted means coiled, and tubule means tiny pipeline or tiny tube. So proximal convoluted tubule is proximity at near to the glomerulus, and distal convoluted tubule is far from the glomerulus. Now CD, collecting duct. The full form of CD is collecting duct. Collecting duct. Okay. Now it's very important to learn about CD. Why it's so? Though we are considering collecting duct as a part of renal tubules, but in a deeper manner, in a deeper meaning, collecting duct, one collecting duct is shared by many nephrons. One collecting duct is shared by many nephrons, right? So if you start the nephron with glomerulus, then Bowman's capsule, then PCT, then LOH, then DCT of multiple nephrons will be connected to the collecting duct. So collecting duct is not nephron's own part. If you are belonging to everybody, then you are belonging to nobody. You are part of nobody. So that's what I wanted to say. So collecting duct is not nephron's own part. Collecting duct is not nephron's own part. Why it's so? This is Bowman's capsule, this is PCT, this is loop of Henle, this is DCT. Now this DCT is connected, the DCT is connected to this duct. Another DCT of another nephron is also getting connected here. Another DCT of another nephron is also getting connected here. So that's why we are saying that CD, collecting duct, is common passage of DCT of multiple nephrons. So collecting duct is a common drainage passage for DCT of multiple nephron. This is DCT of this nephron, this is DCT of another nephron, this is DCT of another nephron. Remember it. So if you're asked which of the following part is not belongs to a nephron's own, so and the options are PCT, Henle's loop, DCT and collecting duct. The answer will be collecting duct. Remember it. Okay. Got it? So you are done. So here is the you can see the diagram, detailed diagram of the glomerulus and Bowman's capsule is exhibited here. So this tuft of blood capillary is glomerulus. You can observe it clearly how they are forming the bunch or tuft of blood capillaries. So tuft means bunch of blood capillaries. Okay. Now if you look carefully, you can observe here a wider blood vessel is entering from here and a comparatively narrower blood vessel is exiting from here, right? So there is an entry point, there is an exit point of blood. And when this blood is flowing through this circuit, it's producing a blood pressure into the glomerulus region, and that will be helping in the process of formation of urine. Later on we will be studying that. So the blood vessel which is responsible for carrying blood towards the glomerulus, that is called afferent arteriole. Afferent arteriole is a branch of what? Branch of renal artery. Okay. Now if you are taking something with the help of your right hand, and if you are donating something, it's the same thing with the help of your left hand. So the right hand will be your afferent hand and your left hand will be the efferent hand. So the blood fetching blood vessel, which is called afferent arteriole, and obviously that is wider in compared to the efferent arteriole. Efferent, EF or efferent arteriole, that is narrow blood vessel. Now, why it's so? It's a magic of the Almighty. See, you need to create blood pressure, into the wall of these glomerular capillaries. Now, how to create blood pressure? If the similar volume of blood is coming from afferent arteriole and the similar volume of blood is going through efferent arteriole, then there will be no formation of pressure or resistance into these glomerular capillaries. In order to avoid that, the Almighty has made the afferent arteriole as a wider channel. So the more volume of blood will enter through afferent arteriole and less volume of blood, more volume of blood is entering through the afferent arteriole and less blood is going out through the efferent arteriole. So if entry is more and exit is less, so the extra volume of blood will create pressure into this glomeruli or glomerulus zone. So that is the scientific explanation or the biological engineering behind this varying diameter or difference in diameter in between afferent arteriole and efferent arteriole. Got it? Now let's talk about the Bowman's capsule. So what is Bowman's capsule? A double-walled cup-like structure, right? A double-walled cup-like structure is called Bowman's capsule. So here you will find inner wall or the visceral wall, and this is the outer wall or the parietal wall of Bowman's capsule. And the tissue present in both the cases, in both the wall of Bowman's capsule, SSEP means short form of simple squamous epithelium. So the tissue present in Bowman's capsule in both the layers, that is simple squamous epithelium. Now in the inner wall or the visceral layer of Bowman's capsule, the simple squamous cells are modified here, and those modified simple squamous cells are called podocytes. The modified simple squamous cells are called podocytes. Now why podocytes? Because if you observe this cell, you'll find pod like foot like structure. So that's why they are called as podocyte. They're nothing but modified simple squamous means flat cells, right? Okay. Now we'll find gap between them, gap between these podocyte cells. Now they help in the process of filtration process during the time of urine formation, and these gaps are called filtration pore or slit pore. These gaps are called filtration pore or slit pore, and the diameter of the slit pores nearby 25 to 40 nanometer. Okay, the measurement can vary, right? So you got my point. And also these glomerular capillaries, they are also fenestrated. So these are tuft of fenestrated capillaries. Now we'll ask, sir, what is fenestrated capillary? In our body, different kinds of blood capillaries are present. So if you find this kind of capillary without any pore into it, so that will be called as continuous capillary. If you get this kind of capillary where capillaries are made up of again simple squamous cells or flat cells, and the cells have this sort of small openings or pores. These pores are called fenestra. These pores are called fenestra. They are of 60 to 80 nanometer size, and this kind of capillaries are called fenestrated capillary. Obviously, this slit pore and these fenestrated capillaries will be helping in the process of filtration process by purifying, by filtering the blood and formation of filtrate. And this kind of capillaries are called continuous capillaries. Okay. So in higher classes, you will be studying about different types of capillaries, about fenestrated capillary, about continuous capillary, about sinusoidal capillaries. Different types of capillaries are there. So we have only discussed the thing which is relevant to this particular topic. Okay. And one more important thing. One thing is written here, Malpighian body. Now what's that? Together, glomerulus plus Bowman's capsule, glomerulus G plus B, Bowman's capsule, will be forming Malpighian body or renal corpuscle or Malpighian corpuscle. Do not confuse this Malpighian body or renal corpuscles or Malpighian corpuscles with Malpighian tubules. So Malpighian body or Malpighian corpuscles is present or they are part of human nephron, right? the glomerulus and the Bowman's capsule togetherly called as Malpighian body or Malpighian corpuscles. Malpighian tubules are the excretory structure present into insects specifically. Okay. Now we'll be studying in detail structure of nephron. Okay. So let's explore it. So this is renal artery. The branch of renal artery is called afferent arteriole. Bringing blood towards the glomerulus. This is glomerulus. And the exit part, which is a narrow tube, that is called efferent arteriole. Okay. Now this double-walled cup-like structure, that is called Bowman's capsule. Now this highly coiled part after the Bowman's capsule, that is called PCT, proximal convoluted tubule. And this hairpin loop shape of structure, that is called loop of Henle. And the loop of Henle has one descending arm, and one ascending arm. The arm or the limb which is bringing the filtrates downwards, that is called descending limb of loop of Henle, and the arm which is bringing filtrate upwards, that's called ascending arm or ascending limb of loop of Henle. Okay. Now this loop of Henle will join with another coil tube which is in distance from this Malpighian body, that is called DCT, distal convoluted tubule. You can see the convolution, the coiling part, and the end part of DCT is called connecting duct. End part of DCT is called connecting duct, that is connected to a common channel of different DCTs are joining, different connecting ducts are joining. This is called collecting duct. And the wider end part of the collecting duct is also called as duct of Bellini. This is called as duct of Bellini. Okay. So these are all about the renal tubule, the tubular part. Now again come to the vascular part of nephron. Now here we ended with the efferent arteriole, which was bringing out blood from the glomeruli or glomerulus. Now you will find here the branches of efferent arteriole, they are getting narrowed down and surrounding the renal tubule. So these structures are called peritubular capillaries. The branch of efferent arterioles surrounding their coiling or surrounded these renal tubular parts, so we are calling them peritubular capillaries. Peritubular capillary. Don't forget this, peritubular capillaries are nothing but their branch or part of efferent arteriole. Now, few special part of peritubular capillaries which are running parallelly to the Henle's loop, that particular part is called as vasa recta. So vasa recta, they are nothing but they are part of peritubular capillaries running parallelly to the Henle's loop. Parallel to HL or Henle's loop. Got it? HL can also be called as LOH. Either you can call it loop of Henle or you can call it HL. And the blood capillaries which are running parallel to the Henle's loop or loop of Henle, that is called vasa recta. Got it? Okay. Now, vasa recta, why this vasa recta or this peritubular capillaries are present here? Basically, they are donating oxygen here, right? And also this vasa recta and Henle's loop will be helping us to maintain the tonicity or osmolarity into the kidney medullary zone. Later on we are going to study this. And the end part of the vasa recta will be exiting with the help of veins, and these veins will be uh, collecting or joining to the renal vein. So we are done with the structure of nephron. We are done with the structure of nephron. Okay. Now let's talk about the types. We were saying that there are two types of nephron present into our kidneys. So let's demonstrate those types of types of nephrons. So if this is the kidney, and this is separation of cortex and medulla. You know it very well. This part is called cortex. This part is called medulla. Okay. Now, if you will find that the nephrons are located towards the cortex and minimally entering into the medullary zone, they are called cortical nephrons. And if you will find that the nephron is present near to the medulla and maximally penetrating into the inner medullary zone, that is called juxta medullary nephrons. So this particular nephron is example of juxtamedullary nephron, and this particular nephron is example of cortical nephron. Now let's compare them. When you talk about their number in our body, so 80 to 85% of the nephrons, means the maximum number of nephron present into our kidneys, are cortical nephrons. So only 15 to 20% are juxtamedullary nephrons. But remember, we need juxtamedullary nephrons to form the concentrated urine, right? And that is again based upon the situation. The concentration of urine will be changing depending upon the water requirement or the electrolyte balance of our body. So sometimes our body will be excreting out isotonic or hypotonic urine. Sometimes our body will excrete out hypertonic urine compared to our body fluid. So when our body is excreting out or eliminating out hypertonic unit, concentrated urine, then you must need this kind of nephrons, juxtamedullary nephrons. It's because they have longer loop of Henle and well developed vasa recta. Let's talk about that. Okay. So here you can see that loop of Henle, LOH, is short in case of cortical nephrons, where loop of Henle is long in case of juxtamedullary nephron, and they go deep into the medulla. They go deep into the medulla. Now let's talk about the vasa recta. Let's check the vasa recta of this particular nephron. Here you can find there's a cortex and medulla zone is divided. The most of the part of this nephron is present into the cortex. Only a little bit of Henle's loop present into the medulla. Now, the capillaries surrounding this renal tubular part, this is called peritubular capillaries, and the part which is going parallel to the Henle's loop, that is called vasa recta. Vasa recta is either absent, or if present, they are highly reduced in case of cortical nephrons, and that is the reason they cannot take part in concentrated urine formation. Why? We're going to come, well, we'll be talking about the counter current theory of urine formation. Okay, but this here you can see one juxtamedullary nephron. Again, difference of zone between cortex and medulla is prominent here, but it's easy to observe that this vasa recta is going parallelly and deep down along with the Henle's loop. So here you can say that this vasa recta is well developed in case of juxtamedullary nephrons. Okay, so altogether we can understand the difference between the two kinds of nephrons. Now, one more question can be asked, that if we talk about a kidney, let's divide the cortex and medulla zone, and this is the nephron. Okay, so which parts are present into the cortex and which parts are present into the medulla if we talk about a single nephrons? So in cortex and medulla, in cortex glomerulus will be present, in cortical zone Bowman's capsule will be present, in cortex zone PCT and DCT will be present. So glomerulus, Bowman's capsule, PCT and DCT present to the cortex zone, whereas Henle's loop or loop of Henle and CD collecting that, they are present into the medullary zone. Let's move forward. Okay, now this is our topic of urine formation. Let's discuss it. Okay, now one more thing when to learn about the nephron, the structure of nephron. One more thing, it will be help you in case the question is asked, tell about the blood flow inside the kidney. We have studied the passage of urine flow. Now we'll talk about the blood flow for kidney. Okay, so the passage will be like this. It will be coming from heart to aorta. The branch of aorta will be dorsal aorta. The branch of aorta will be dorsal aorta. Okay. Now from the dorsal aorta, the branch that enters into the kidney, that is called renal artery. The branch of renal artery that gets divided inside the tissue of kidneys, that is called afferent arteriole. Afferent arteriole will put the blood into glomerulus. Now when it's exiting from glomerulus, it will become efferent arteriole. From efferent arteriole, the blood will be channelized into peritubular capillaries. From peritubular capillaries to vasa recta, and the small branches of veins will be calling as venules. The venules will be joined into the renal vein. Renal vein will channelize the blood into the prime vein, that is the inferior vena cava. Inferior vena cava, and that will carry the deoxygenated blood and again will be going towards the right side of our heart. So this is the pathway of blood flow for our excretory system. Okay, now let's start about the process of urine formation. Yes, now obviously for that we have efficient biological machineries present inside our kidneys. They are called nephrons, and the process of urine formation involves three main processes. Okay, and they are named as first glomerular ultrafiltration, the first step, the second step reabsorption, and the third step is secretion. The first step is glomerular ultrafiltration, second step tubular reabsorption, the third step is tubular secretion. Okay, now what happens basically for these things? When we talk about glomerular filtration, when we talk about glomerular filtration, blood which is passing through the glomerulus will be filtered out the junction of glomerulus and Bowman's capsule and get inside the lumen of Bowman's capsule, and we will be calling this as filtrate. Right. So this process is basically called glomerular filtration or glomerular ultrafiltration. Okay. Now why it's called glomerular ultrafiltration? Why it's called glomerular ultrafiltration? Because all the component of blood is getting filtered out except the formed elements of the blood cells and proteins. That's why the process is called as ultrafiltration. All the components of blood is getting filtered except formed elements of blood, means blood cells, and proteins. That's why the process is so much fine that it won't be filtering out the proteins and the blood cells. That's why you are calling this process as ultrafiltration. Okay. Now, one question can be asked that how much volume of blood is passing every minute through this circuit of afferent arteriole, glomerulus, and efferent arteriole of kidney. So that is called as renal blood flow, RBF, renal blood flow. The volume of blood is passing by kidneys in 1 minute. That is called renal blood flow, and the volume will be 1100 to 1200 ml. That is called renal blood flow. Okay. Now, if we talk about the blood pumping capacity of the ventricle, the left ventricle of your heart, that is in a single stroke, your one ventricle is pumping out 70 ml of blood, and such kind of 70 to 75 strokes will be there, and that is called cardiac output. So in a minute, the left ventricle, in a minute, the left ventricle of your heart is pumping out near about 5 to 5.5 liters of blood into your arteries. So that is called cardiac output. cardiac output of our body is nearby 5.5 liter, or you can say 5 to 6 liter, right? Or 5 liter in lump sum, you can say the cardiac output will be 5 liter or 5000 ml. Now, if you compare both the data, renal blood flow and cardiac output, then easily we can say that renal blood flow is 1/5 of cardiac output. Renal blood flow is 1/5 of cardiac output. Cardiac output is near about 5 to 6 liter, and renal blood flow is only the 1/5 part of 5000 ml or 6000 ml. So, it can be said like this: RBF, renal blood flow, is 1/5 of cardiac output. Remember this. Now, if someone asks you, bro, tell me about renal plasma flow, focus properly. If renal blood flow is 1100 to 1200 ml per minute, then in 1 liter blood, how much plasma, or what will be the volume of plasma present in 1 liter blood? 55% of volume of blood is plasma. So, if the blood quantity, the volume of blood is 1 liter, so the plasma will be near about 550 ml. And if the blood volume is 1200 ml, so the plasma flow will be near about 600 to 625 ml. So, that will be called as renal plasma flow. So, whenever you are writing, if you are asked that how much blood is getting or flowing through this circuit of afferent arteriole, glomerulus or efferent arteriole in all the circuits of kidney in 1 minute. So, the volume of blood will be 1100 to 1200 ml, that is renal blood flow. And if you are asked what is RPF, renal plasma flow, just you have to take out the 55% of this 1100 ml or 1200 ml per minute. Now, if you are asked that it's fine, 1200 ml of blood is flowing through this circuit, but what amount of blood is getting filtered out? How much filtrate is produced? That is called GFR, glomerular filtration rate. So, if you search for the answer, the filtrate production will be, it's like that, 125 ml of filtrate will be produced per minute, right? And that is called as glomerular filtration rate, GFR, glomerular filtration rate. That is what amount of filtrate is produced per minute. So, if in a minute 125 ml of filtrate is produced, that is GFR per minute. Now, if you are asked what is GFR in a day, so the answer will be 180 liter. So, the GFR per minute is 125 ml, but if you convert it into a day, so it will be 180 liter in a day. So, in both the cases, question can be asked, the GFR for a day or the GFR for a minute. Got it? Okay. Now, if you understand this, we'll be moving forward. The next step, that is reabsorption, we'll be briefing it. We are coming with in detailed explanation of glomerular filtration. Now, what is reabsorption? So, you know that our nephrons are surrounded by blood vessels. Nephrons are surrounded by blood vessels. By peritubular capillaries. So, there will be always exchange of materials in between the blood and the renal tubules. So the reabsorption means exchange of material in between renal tubule and blood. So that is called reabsorption. So reabsorption means materials are flow of materials from renal tubule, means from PCT, Henle's loop, DCT to blood vessels. So this simple process is called tubular reabsorption. Blood is reabsorbing things from renal tubules. Now why it's so? Once blood is getting filtered into the in between the glomerulus and Bowman's capsule, and inside the Bowman's capsule the filtrate is formed, body is observing that this filtrate is containing many important components into this, like it has glucose, it has amino acids, it has too much amount of water, it has too much amount of electrolytes. So if you excrete out all the things with the help of nephron, then very soon the body will be getting exhausted. It's something like that. Suppose there is a program in your home, your relatives are coming. Now your mom suddenly entered into your particular room, into your study room, and she observed that your study room is messy. Now she became angry. She threw all the things. Upon anger, she threw all the things from your room to outside. Now after that, she observed that, she felt that, okay, upon anger I have eliminated all the things from my son's or from my daughter's room, but there are some important books are there, NCERT books are there, which is important for his or her NEET exam. So let's bring all the things again, or the necessary things again, and all the waste material eliminate out. So our body also does the same thing. When it's doing the filtration process, glomerular filtration, it will be filtering out some important materials as well in that form of filtrate. But as soon as body realize, when the filtrate is passing through the PCT, Henle's loop and DCT, the body realizes that this filtrate is containing some important material like glucose, amino acids, some other nutrients, some salts, some electrolytes, some water. So body will take them back. So that process is called reabsorption. Now what is secretion? Secretion is the direct removal of some unwanted material, for like body will be secreting hydrogen ions or protons, body will be secreting ammonia directly into the renal tubules, body will be secreting potassium directly into the renal tubules. That is called selective secretion. Secretion means selective release of protons, H plus ion, potassium ion, ammonia from blood vessels to renal tubules. That is called the process of secretion. So reabsorption and secretion, they are opposite phenomena. Again I am simplifying it. If this tube is renal tubule and this tube is blood vessel, then if materials are flowing from renal tubule towards the blood vessel, that is called the process of reabsorption. And if materials are flowing from blood vessel towards the renal tubule directly, that will be called as the process of secretion. Now let's move forward. Okay, now we'll be elaborating the process of ultrafiltration. Okay, so we already discussed this, what is glomerular ultrafiltration, how much blood will be crossing this circuit of glomerulus, afferent arteriole, glomerulus and efferent arteriole, that is renal blood flow. Renal blood flow is about 1100 to 1200 ml per minute, and how much filtrate will be produced per minute? 125 ml filtrate will be produced per minute. So the liquid produced initially inside your nephron, that is called as filtrate. One very important thing about this, inside PCT we'll be calling the fluid as filtrate. Inside Henle's loop, inside DCT we'll be calling the fluid as filtrate. When the fluid will inside the nephron, when the fluid will enter into the collecting duct, then only we can call it urine. So inside the collecting duct the fluid is present, that is called urine. Before collecting duct, inside Bowman's capsule, inside PCT, inside Henle's loop, inside DCT, the fluid is called as filtrate only. Okay. And if you are asked what is the tonicity or osmolarity of the filtrate, if you compare with your blood or with your plasma, so the osmolarity or the tonicity of your plasma is 300 milliosmol per liter, and the tonicity of the osmolarity of filtrate is similar, 300 milliosmol per liter. So initial filtrate and the plasma is isotonic in nature. The concentration of our blood or plasma is equal to the concentration of produced filtrate. But the urine we are going to produce here, we have to produce 4 times more concentrated urine, means ultimately the urine will be passing out to reserve water into our body. The concentration of the urine is going to be hypertonic, remember. But the filtrate initially that is produced, that is isotonic. That is isotonic in compared to whom? In compared to the tonicity or the osmolarity of our blood or plasma. Got it? Okay. Now here everything is mentioned here properly. In order to facilitate the process, in order to make the process easier, the glomerular capillaries has tiny pores. We call them fenestra, and the fenestra are average 70 nanometer size, and these are the podocyte cells. And in between podocytes you can find gap is here, so that gap is called as filtration slit, and this filtration slit are also can be called as slit pore as written in NCERT. Their diameter or their size is in between somehow 25 to 40 nanometer, 25 to 40 nanometer. Okay. Now obviously the blood cells like RBC, WBC, they are quite larger than this, so they cannot pass these pores, right? Even few proteins, most of the proteins cannot pass this process, cannot pass this particular barrier. Why proteins cannot pass? Proteins, the size of proteins are even smaller, like the size of albumin is smaller than the fenestra or the filtration slit. But the albumin, the most abundant protein into our plasma, cannot pass this barrier. Why? Because in between the glomerular wall and the Bowman's capsule wall, you will be finding a basement membrane, and the proteins associated with the basement membrane, that is negatively charged. That is negatively charged, and our albumin proteins are also negatively charged. So that's why negative will repel the negative, so they cannot cross this barrier. Now, obviously, while the blood is getting filtered out from glomerulus to Bowman's capsule lumen, it has to cross barriers. Now let's talk about those barriers, those trilaminar filtration. We call this process of filtration as trilaminar filtration as well. Now why trilaminar filtration? If your blood, you have to cross 3 layers. What are those layers? The first layer, that is endothelium, the wall of these glomerular capillaries. Endothelium of glomerular capillary. This is the first layer. The second layer present in between the Bowman capsule and the glomerulus, it is called basement substance or basement membranes. Basement membrane made up of basement substance. And the third layer that is called epithelium of Bowman's capsule. So while blood is getting filtered, it has to cross endothelium of glomerulus, basement membrane, epithelium of Bowman's capsule, and then only is getting converted into filtrate. That's why you can call the process as well as glomerular ultrafiltration as well as uh, trilaminar filtration. Trilaminar filtration. Got it? So we know about GFR, we know about renal blood flow. Now we need to know about the forces. We need to know about the forces or osmotic pressure which are behind the process of glomerular ultrafiltration. Let's check them. Okay, so three forces are acting here. Three forces are acting here which are helping or facilitating the process of glomerular ultrafiltration. So again, I will draw this Malpighian body part, means Bowman's capsule, and the glomerular part, afferent arteriole, glomerulus, and efferent arteriole. Okay. Now the blood is flowing. When the blood is producing pressure due to the difference in the diameter of afferent arteriole and efferent arteriole, so the pressure is produced, that is called uh, glomerular blood pressure, right? Or you can say glomerular hydrostatic force. Glomerular blood pressure or glomerular hydrostatic force. Just a minute. Okay. Glomerular hydrostatic force or glomerular hydrostatic pressure. The pressure of blood towards this direction, which is acting from glomerulus towards the Bowman's capsule lumen. Okay. And two forces are acting opposite to this. Who are they? One force is acting from Bowman's capsule towards the glomerular capillaries. So name of that pressure is BCOP, blood colloidal osmotic pressure. Now what is blood colloidal osmotic pressure? There will be presence of colloid particles or proteins like albumins present into the blood. Albumin proteins are present into the blood, and they have a tendency to retain water or to attract water from Bowman's capsule towards the glomerular capillaries. That is called blood colloidal osmotic pressure. So that's why you have studied in the chapter, in another chapter, this body fluid and circulation, albumin, the most abundant plasma protein that helps us to maintain the osmolarity or osmotic pressure of our body. Okay, so this GHP has a tendency to release water from glomerulus to Bowman's capsule, but BCOP, blood colloidal osmotic pressure, has an opposite tendency to retain water. Okay. Now, once filtrate formation will be started into the Bowman's capsule, the filtrate also has a lot of water, near about 90% of the volume of the filtrate will be water only. So again, that will create a hydrostatic pressure from Bowman's capsule to the glomerular side, and that is called capsular hydrostatic pressure. Capsule hydrostatic pressure, CHP. So here three forces are acting. The yellow line, that is GHP, glomerular hydrostatic pressure, and the blue arrow and the green arrow, they represent capsule hydrostatic pressure and blood colloidal osmotic pressure, and you can observe it here clearly that GHP, the direction of the action of GHP is opposite to the direction of the combination of BCOP and CHP. And with this calculation, we can calculate the net effective filtration pressure. Net effective filtration pressure. So, how to calculate net effective filtration pressure? We have to take GHP, glomerular hydrostatic pressure, and we have to minus BCOP plus CHP, capsular hydrostatic pressure, from the glomerular hydrostatic pressure. The average glomerular hydrostatic pressure, suppose it's 60 mm mercury pressure, the average BCOP is going to be 30 mm mercury pressure, 30 to 35, and the average CHP is going to be 15 mm mercury pressure. Now, if you calculate this in this manner, 30 plus 15, 45, and 60 minus 45, 15 mm Hg. So, this is the net effective filtration pressure. So, ultimately who wins? Glomerular hydrostatic pressure wins, because it is more than the summation of BCOP and CHP, which are acting from Bowman capsule to the glomerulus, but the GHP is acting from glomerulus to the Bowman's capsule, but the GHP is acting towards the Bowman's capsule and it wins, and that causes the formation of filtrate into the Bowman's capsule. So, this sort of calculation can be asked. Okay. Now, for this we have to give thanks to the diameter range of afferent arteriole and efferent arteriole. Okay. Now, the next thing, NCERT says that in certain condition, like if your body face high BP, high blood pressure, hypertension, or hypotension, low BP, low blood pressure, in that case, or if you are not drinking enough water, in that case, the GFR may fluctuate, the GFR may change, but our body always tries to maintain the constant GFR of 125 ml filtrate production per minute, and if that get increases or decreases, that will affect ultimately our kidney health or kidney function, and that may cause kidney failure, right? If GFR get increases or GFR get decreases, that can affect kidney function or urination output, and also in long term can cause a kidney failure, and if the kidney is failed, it's going to affect numerous activities of our body. It's going to affect our blood pressure, it's going to affect our heart and other body functions, right? So, now let's talk about that, how body are managing, how our body is managing those fluctuating GFR. Now, our body has inbuilt mechanism to fix fluctuation or fix change in GFR. What are the conditions that can change GFR? If there is high blood pressure, the GFR will increase. If there is low blood pressure into your body, the GFR will decrease. More blood flow means more output, less blood flow means less output. I said both the conditions are not good. So, how our body is going to manage these conditions? So, our body has inbuilt mechanism. First thing, our body will utilize JG apparatus to measure all these changes, to measure the blood flow inside the kidney, to measure the filtrate flow inside the kidney, to measure the concentration of the fluid. We have sensors present into our kidneys, and those sensors are called JGA, juxtaglomerular apparatus. Now, what is JGA? We'll be discussing about that. The second mechanism our body has, the kidney has, that is called myogenic mechanism. Have special muscle cells, special smooth muscle cells present into the wall of the afferent and efferent arterioles. They can alter the blood pressure, and ultimately blood pressure is altered, GFR will be altered. That is called myogenic mechanism. So myogenic means where some smooth muscles are responsible. Where you will be getting these smooth muscles? Smooth muscles of AA means afferent arteriole and efferent arteriole. Okay, they can sense the change in blood pressure. Okay, the third mechanism is a cascade, that is called RAS system. Renin, angiotensin, aldosterone system. Right. We have a special hormone, ADH, antidiuretic hormone, that can alter or fix the rate of GFR if necessary. We have atrial natriuretic factor that can sometime, depending upon the situation, it can also exhibit its effect towards the fixation of altered GFR. So our body has all this mechanism to fix the clock of our kidney in a correct manner. Now I was talking about JGA, juxtaglomerular apparatus. If this is the nephron, this is the afferent arteriole, and this is the efferent arteriole. At the junction of this DCT and afferent arteriole, at the junction of this DCT and the afferent arteriole, we get a special device, a specially designed device, that is the JGA, juxtaglomerular apparatus. Apparatus means obviously device. Juxta means near, to glomerulus, that's why you are calling it juxtaglomerular apparatus. Juxta means near. So this device present in between the wall of afferent arteriole, because the cells of the walls of afferent arteriole and DCT are getting modified to form this device. So you are calling this as JGA. We are calling this apparatus as JGA, juxtaglomerular apparatus. Now if you observe it carefully, you will be finding the presence of three types of cell. The presence of three types of cell present in between into this JGA. What are those three types of cells? First, towards the afferent arteriole, the cells are called JG cells, juxtaglomerular cells. In between juxtaglomerular cells and this densely packed cells, you will be getting lattice cells or Pulkyson cells, and the cells present towards the DCT, the third layer, it is called macula densa cells. Macula densa cells. The densely colored cells are called macula densa cells. Okay. Now, what are the importance of these cells? If we talk about JG cells, juxtaglomerular cells, they are responsible for production of renin, This is hormone cum enzyme. Do not confuse with RENNIN. There is another renin, RENNIN, that is an enzyme. But RENIN, that is hormone cum enzyme. JG cells also can manage the RBC forming hormone production. It is called also precursor cells of erythropoietin. JG cells also will be producing renin as well as erythropoietin. Erythropoietin, they are responsible for RBC production. Okay. Macula, and one more thing about JG cells, they can feel the pressure of your blood, so they act as baroreceptor. Whereas these macular densa cells, they can sense the osmolarity of the fluid passing into the distal convoluted tubule. So this macula densa cell, they act as osmoreceptors. And if there is a change in the osmolarity, the macula densa cell will send signal to JG cell with the help of the Lassie cell. So that's how the GFR will be fixed with the help of juxtaglomerular apparatus. Okay. Now we are done with ultrafiltration process. Now we'll talk about the reabsorption and secretion. So as we have seen that if we calculate the total GFR of our body per day, that will be 180 liter of filtrate is produced in a day. But if we talk about the urine is formed in a day, that then it will be near about 1 to 1.5 liter a day. So that means though our kidneys are producing 180 liter of filtrate, is obviously it's filtering the same blood again and again. So that's why the summation of the filtrate production will be 180 liter, right? But only excreting out 1 to 1.5 liter in a day. That means 99% of the filtrate is getting reabsorbed. So this is very much important. 99% of the filtrate is getting reabsorbed. Who is reabsorbing them? Blood, obviously. Who is reabsorbing them? Blood is reabsorbing them. Now we have to study the different parts of nephron are involved in the process of reabsorption, and also we'll talk about the secretion or selective secretion of different ions or minerals with the help of different parts of nephron or renal tubules. Okay. So with this diagram, you can observe this diagram in your NCERT as well. So first, the filtrate is getting produced into the Bowman's capsule, then it's coming into the PCT, proximal convoluted tubule. Now, you need to learn this particular information that the highest rate of reabsorption will be taking place through proximal convoluted tubule. highest rate of reabsorption. The next slide will be writing this. So just for the sake of information, I am telling you PCT, for the PCT there will be highest rate of reabsorption will be taking place, and what are the components will be reabsorbed by through PCT. All the nutrients present into the filtrate, it is 100% of the nutrients. If you talk about nutrients, there will be reabsorption of glucose, 100% of glucose, 100% of amino acids, okay. There will be reabsorption of salt as well as water. Now remember this, that a similar concentration of salt means NaCl and water will be reabsorbed. Near about 70 to 80% of salt as well as water will be reabsorbed through PCT. And if you are asked why PCT shows highest rate of reabsorption, you have to tell about the nature of the tissue present into the inner lining of PCT, that is brush border cuboidal epithelial tissue. They are expert in reabsorption. Which tissue? Brush border cuboidal epithelial tissue. In PCT, the texture of cells will be like this: cuboidal cells with microvilli. That's why they will exhibit highest reabsorption. There will be reabsorption of bicarbonate ion HCO3 minus. There will be reabsorption of potassium ion K plus. Now, let's come to the Henle's loop. First, we will talk about the descending limb of loop of Henle. Now, the nature of tissue is present into the descending limb. Now, when you are observing this descending limb and ascending limb, both of them have a thick part and a thin part. Thin part and a thick part. The thick part has the simple cuboidal epithelial cells, and thin part has squamous epithelial cells. Thin part has squamous epithelial cells, thick part has simple cuboidal epithelial cells. Okay. Now for descending limb, there will be reabsorption of water molecules, which is permeable to water but impermeable to salts and electrolytes, the descending limb of loop of Henle. But when you talk about ascending limb of loop of Henle, this is permeable. There will be reabsorption of sodium chloride, means salt. This is permeable to salt and electrolytes but impermeable to water. What? Ascending limb. And the rate of reabsorption is lowest. Rate of reabsorption is lowest if we talk about ascending limb. Rate of reabsorption is highest in PCT. Rate of reabsorption is lowest in ascending limb. Okay. Now through ascending limb is which component is getting reabsorbed? Salt. Na plus, Cl minus as well as potassium ions. Okay. Now, when you talk about the reabsorption of electrolytes through the ascending limb, it may follow the active transport or passive transport. The thin segment of ascending limb will reabsorb, there will be reabsorption of salt into the passive transport mechanism. Passive reabsorption will be there through the thin segment, and with the help of thick segment there will be active reabsorption of salt when you are talking about ascending limb. Okay. Now DCT, there will be conditional reabsorption, not always, if required, conditional reabsorption of salt and water, that is influenced by the hormone called aldosterone. There will be reabsorption of bicarbonate ion. Now let's come to collecting duct. Through collecting duct, there will be reabsorption of water molecules. Through collecting duct, the distal, especially through the distal part of collecting duct, there will be reabsorption of urea. We are done with reabsorption. Now let's talk about the secretion. Now let's talk about the secretion part. Come to PCT, there will be selective secretion of proton in H plus ions and ammonia NH3. There will be no secretion in case of Henle's loop, but with the help of thin segment of ascending up, the urea will enter into this ascending limb of Henle's loop. If we talk about the secretion in DCT, there will be selective secretion. There will be selective secretion of ammonia, potassium, and H plus ion. Now here you need to remember this: potassium is getting reabsorbed through PCT, but it's getting secreted through DCT. Now if we talk about the secretion in collecting duct, there will be secretion of potassium ions and H plus ions, right? Now in NCERT, a separate line is given for the mode of transportation of all these materials. So we know that mode of transportation can be of two types: active transport and passive transport. When there will be utilization of ATP for the mode of transportation, then that is called active transport, and if no ATP is needed for the mode of transportation, that is called passive transport. Yes. Now for active transport and passive transport, if we compare, what are the components which will be actively transported either during the time of secretion or absorption? They are sodium ion, potassium ion, nutrients like amino acid, glucose, bicarbonate ion HCO3 minus, and protons or H plus ions. Now if we talk about the passive mode of transportation during the time of reabsorption or secretion, nitrogenous wastes will be transported passively. Transport of water through the process of osmosis, that is again a kind of passive transport, and transport of chloride ions, Cl minus ions, will be through passive manner. Okay. Now in some special case, sodium chloride transportation may happen passively when I am talking about this thin part of ascending limb. Here the reabsorption or the transportation of sodium chloride will be happening passively, not actively. You need to remember this. Now let's make a chart. All the parts of renal tubules and their specific reabsorption and secretion. Okay. So first talk about PCT. It shows highest rate of reabsorption. An ascending arm of loop of Henle shows lowest rate of reabsorption. Now, component wise, PCT will be reabsorbing nutrients, 100% of the glucose and amino acids, 70 to 80% of salt and water. There will be reabsorption of potassium ion, there will be reabsorption of bicarbonate ion through PCT. Now, if we talk about secretion in PCT, these components will be secreted into PCT. Ammonia will be secreted into PCT, and H plus ion will be secreted into PCT. For descending arm of loop of Henle, there will be reabsorption of water. You need to remember this. This part is impermeable to electrolytes. There will be no reabsorption or transport of salt if you are talking about the descending arm of loop of Henle. Again, there will be no secretion taking place inside the descending limb. For the ascending arm or ascending limb of loop of Henle, there will be secretion of sodium chloride. And again, you need to know this part is again opposite to the descending arm. Ascending arm is impermeable to water. Now, if someone asks you question, why descending arm is permeable to water and why ascending arm is impermeable to water, you have to answer with the reference of the presence of particular tissue. The most of the part of descending arm you will be getting made up of simple squamous epithelial tissue. Most of the part of ascending arm, it is made up of a simple cuboidal epithelial tissue. And obviously, if different tissue cells are present, their transporting proteins will be different. So that's why their transportation capability will be different. Their permeability will be different. Now, if you're talking about secretion through ascending limb of loop of Henle, yes, there will be secretion of urea through the thin segment of ascending limb of loop of Henle. Now, let's come to the DCT per distal convoluted tubule. What are the substances will be reabsorbed through DCT? Conditional reabsorption of water. Conditional reabsorption of, first there will be reabsorption of salt and then there will be reabsorption of water. Water will be following the salt. So, when you are talking about DCT or PCT, first there will be reabsorption of salt and then there will be reabsorption of water, right? For DCT, it is based upon condition. If aldosterone will stimulate the lining of cells of DCT, then only there will be reabsorption of salt, and first salt, then water will be following the salt. And also there will be reabsorption of bicarbonate, HCO3 minus. Now, let's come to the secretion into the DCT. What are the components will be secreted into the DCT? Ammonia will be secreted into the DCT. H plus ion will be secreted into the DCT. K plus potassium ion will be secreted into the DCT, unlike the PCT. Now, let's come to the CD, collecting duct. Though it's not a part of nephron, remember this. The things will be reabsorbed here, that is majorly water and urea through the distal part. Distal part of CD is responsible for mainly reabsorption of urea. Okay. Will there be secretion in CD? Yes, there will be secretion of potassium ion, there will be secretion of H plus ion in case of collecting duct. So, this is how our urine is formed by following the 3 steps. There is glomerular filtration, then reabsorption, then the selective secretion. Now, we are mammal. We have a special capability to produce concentrated urine, right? We are not aquatic organisms, or the animals who are mainly dwelling in land or salt water, marine water, they need this capacity to produce concentrated urine in terms of in order to conserve water into our body, to minimize the loss of water. Though we are ureotelic, but we have to make a reserve, conserve water inside our body, and for that we have a special mechanism of concentration of the filtrate. Ultimately, we will be producing 4 times more concentrated urine compared to our body fluid. Okay, so let's talk about that. So it's written here, mammals have special ability. Mammals have special ability to produce concentrated urine, and the mechanism is called as counter current mechanism. The mechanism is called counter current. Now, what do you mean by counter current mechanism? If opposite directional flow of fluid is there, if opposite directional flow of fluid is there, then that particular mechanism is called as counter current, right? The counter current mechanisms that is involved in this process of concentrating the filtrate in our kidneys. In fishes, the counter current mechanism will act between their gills and water to help their breathing process or gaseous exchange process. So, in our body, different types of counter currents can be seen, but this particular counter current mechanism is for to produce concentrated urine. And the parts involved in this counter current, that is Henle's loop, a loop of Henle, and vasa recta. These two are the parts which are responsible to produce this counter current. Okay. Now, what is the aim of this counter current? Why this Henle's loop and vasa recta will walk together? They are basically producing a corticomedullary, or you can say corticopapillary concentration gradient. So, it's like that. If you observe a healthy kidney, and if you take one part of it, this is the cortex, this is the medulla. So, you can observe towards the cortex, the concentration of fluid present into it of 300 milliosmol. Towards the medulla, inner medulla, the concentration will be 1200 milliosmol. It's gradually increasing from 300, 400, 500, then 700, 800, then 900, then 1000, then 1100, then 1200. So, from cortex towards the inner medulla, gradually increasing concentration can be found, and that is called corticomedullary concentration gradient. That is the nature of a healthy kidney. Okay. Now, what is the aim? Why our kidney is maintaining this concentration gradient? Gradient means towards the cortex, the osmolarity हेलो चेक उसमें नहीं आया था क्या ओ, मतलब उस रिकॉर्ड नहीं हुआ क्या कोई नहीं ठीक है so towards the cortex can be said as hypotonic or hypoosmolar, and the medulla can be said as hypertonic or hyperosmolar. So from hypotonic to hypertonic, that's the charisma of the gradient formation. And why the gradient is formed? What the kidney is aiming to do? When the concentration gradient will be formed and maintained, not only formation of the concentration gradient, you have to maintain the concentration gradient. When you are maintaining the concentration gradient, ultimately your collecting duct will allow easy passage of water to reabsorb, and from collecting duct, if water is getting reabsorbed, after at the end of collecting duct, you will be getting concentrated urine. So the whole sole aim of this process is to produce the concentration gradient, and what that will do? That will do easy passage of water to reabsorb through collecting duct. As simple as that. Again, I am drawing a diagram of nephron. Okay, so the funda is something like this. Above this, everything is cortex. Below this, that is medulla. This is upper medulla, this is the inner medulla. And the space in between this is called interstitial space. Where you will be getting the tissue fluids, and mainly our kidney will be forming this concentration gradient into this interstitial space. To the cortex, the concentration will be 300 milliosmol per liter. It is equal to our body fluid 300 milliosmol per liter. Now to make it very clear, I am saying that suppose you take 1 liter of water, and inside that water you are dissolving salt into it, and suppose there are 300 millions particles of salt or solute particles are there. So you will be calling that concentration of that particular solution 300 milliosmol. Now if you add more salt, suppose now if you count inside 1 liter of water, if you get 1200 millions particles of salts are present, so you will be calling the tonicity or the osmolarity of that solution will be 1200 milliosmol. So 1200 is called hypertonic here, and 300 will be called as hypotonic in compared to that. But the 300 milliosmol, again the osmolarity or the tonicity of our blood and the osmolarity of our plasma. And towards the inner medulla, the osmolarity is 1200 milliosmol, and gradually it is increasing from 300 to 400, 500, 600, 700, 800, 900, 1000, 1100, then 1200. So that is the osmotic gradient or concentration gradient. So ultimately what will happen, the filtrate from DCT will enter into the CD, and that filtrate inside the CD, that is called urine. Urine will coming the downstream downwards, and when the urine or the hypotonic urine will observe the concentration gradient, it will try to follow the concentration gradient of the interstitial space, and to match the concentration gradient it will start releasing water particles into the interstitial space, and that water will get inside the blood vessels, and gradually inside the collecting duct the concentration of urine will start increasing, and at the end, while the urine is released from the collecting duct, the concentration of the urine will become 1200 milliosmol, equal to the inner medullary zone, and that is called hypertonic urine or concentrated urine, which is almost 4 times to our initial filtrate or to our body fluid. 300 into 4 equals to 1200. Okay. Now the question is asked, everything is fine, but which are the substances responsible for making the hypertonicity or hyperosmolarity of the inner medullary zone? So the gradient causing agents are basically salt, sodium chloride, and also urea. They are the gradient causing components. And after that, 4x concentration, 4 times the urine which will be produced, that will be 4 times more concentrated in compared to the initial filtrate or in compared to our blood. Now let's talk about the counter current mechanism with proper diagram. This is the Henle's loop, descending arm, ascending arm, and the parallel blood vessel vasa recta. Right? So the white one, that is the Henle's loop, and the red one, that is the vasa recta. This is the descending arm of Henle's loop. This is the ascending arm of Henle's loop. Now already we know that the person is having healthy kidney. And the concentration is like this. Towards the cortex is 300, towards the medulla it's 1200, and gradually it's increasing 300 to 400, then 500, 600, 700, 800, 900. Thousand, 1100, and ultimately 1200 towards the inner medulla. But the fluid which is entering into the descending limb from PCT, that also has the isotonicity with the fluid, that is 300 milliosmolarity. So the filtrate which is entering into the descending limb having osmolarity of 300 milliosmol. But as soon as it will coming downwards, what it will observe, the surrounding interstitial fluid is getting hypertonic. Now the fluid or the filtrate inside the descending limb will try to manage, try to equalize the concentration with the surrounding osmolarity. So for that, what it can do, it has to increase the osmolarity if it's going downwards. So to increase osmolarity, you have two options. If you have a solution, either you can add more solute into it to increase the osmolarity, or you have to remove the solvent particles to increase the osmolarity. So how, which solution is called more concentrated? The solution which has a higher ratio of solute than solvent. So what property that descending limb is pausing? Is it possible to add more solute into the descending arm? No, because this is impermeable to salt, impermeable to electrolyte, but it's permeable to water. So what it will do, it will start reabsorbing, start throwing out water molecules. So when it's losing the water molecules, which is the solvent, and it's coming downwards, it will start increasing the osmolarity and will start matching the surrounding interstitial fluid. And when it's coming this U-turn, U-loop, this loop position towards the inner medulla, it will almost match the osmolarity with the inner medullary concentration, that is 1200 milliosmol. So when you're talking about the descending limb of loop of Henle, descending limb of loop of Henle, this is called the concentrating limb. The fluid is getting concentrated downwards in descending limb. But when we'll talk about ascending limb, we'll see gradually the fluid is going upwards, will be losing the concentration, and that's why we call the ascending limb as the deconcentrating or diluting limb. So the fluid or filtrate is getting hypertonic in descending limb, and the fluid will become hypotonic inside the ascending limb. Why it's so? After the U-turn, when the fluid is going upwards through the ascending limb of loop of Henle, it will again observe the surrounding osmolarity is becoming hypotonic, going from higher to lower. So what it will start? It will start releasing salt. it will start releasing salt particle to make the solution hypotonic. So the filtrate which is entering from ascending limb to the DCT, that will again become hypotonic with the osmolarity of 300 milliosmol. So that's how is the Henle's loop, is the HL Henle's loop, which is responsible for making the concentration gradient from 300 to 1200. So that is called as counter current multiplication. The osmolarity of cortex and medulla, if you compare it, the medullary osmolarity is 4 times more in compared to the cortical osmolarity. So the osmolarity is multiplied by 4 times inside your kidney. It is called counter current multiplication, and the mechanism which this Henle's loop is playing, that is counter current multiplier. So Henle's loop is responsible for formation of this concentration gradient. Henle's loop is responsible for the formation of this concentration gradient from 300 milliosmol to 1200 milliosmol from cortex to medulla. Okay. Now, what is the role of vasa recta then? Vasa recta, that is responsible for maintaining the osmolarity. So, we will be calling this process which is played by vasa recta counter current maintainer. Multiplication of osmolarity will be done by Henle's loop, and the maintenance of this continuous, that's constant gradient, will be done by vasa recta. So how vasa recta will work now? See, so we can say that if you compare the descending limb and ascending limb, the flow, the directional flow is into the opposite direction. So that can be called as counter current of the filtrate, counter current of the filtrate into the Henle's loop. But there will be flow of blood into the opposite direction into two limb of vasa recta. Now, parallel to the ascending limb, the limb of vasa recta, that is the descending limb, because that is flowing down the blood. So parallel to the ascending limb of Henle's loop, we got the descending limb of vasa recta, and parallel to the descending limb of Henle's loop, we got the ascending limb of vasa recta, vice versa. Again, the blood is flowing down through the descending limb and the blood is flowing up through the ascending limb. So again, in the vasa recta, there will be counter current of blood. And the counter current of blood is also responsible for exchanging the excessive electrolytes and water. So that's why vasa recta can also be called as responsible for counter current exchanger. That's how it's maintaining the concentration gradient. If you delete vasa recta from here, if you give free hand to this Henle's loop, then continuously it will keep increasing the osmolarity of the kidney interstitial space. So everywhere there will be too much amount of salt inside the kidney. Everywhere the interstitial fluid will become 1200 milliosmol, will gain the osmolarity 1200 milliosmol. So the kidney will become hypertonic or hyperosmolar. But if it's gone so, kidney will lose the concentration gradient or corticomedullary concentration gradient. Now you have to maintain it. How you can maintain this? By exchanging the excess salt and water. Now who will be exchanging the excess salt or water? The blood flowing into the counter current direction inside the vasa recta. So here when blood is coming down parallelly to the ascending limb, what it will do? It will start intaking the salt, excess salt which is released by the ascending limb, and the blood which is entering with 300 milliosmol. By gaining salt, it will gain the osmolarity, it will become hypertonic. So gradually it will become 400 milliosmol, 500 milliosmol, 600 milliosmol, 700 milliosmol, because it's gaining more and more salt from the surroundings. The excess salt which is released by this ascending limb, and by the time when the vasa recta, blood inside the vasa recta is coming to the loop, to the junction, it will become 4 times more hyperosmolar than the initial blood. That also gain 1200 milliosmol per liter osmolarity. Again the opposite thing, when the blood is following the ascending limb and going upwards, by observing the outside interstitial concentration gradient, it will try to follow the same. It will release the extra salts which it had absorbed. So it will start releasing salts here, because when it's going upwards, it has to become hypoosmolar. And how it can become hypoosmolar? The blood, by releasing extra salt. Suppose first you have started eating. Now your stomach is full. Now what will happen? You will start vomiting. The similar thing happens with the blood inside the vasa recta. So in descending arm, while the blood is coming down, it will start intaking the excess salts which are reabsorbed, which are released from the ascending limb, and after gaining salts it will become hyperosmolar. And when the vasa recta, the blood inside the vasa recta is following the ascending limb of vasa recta, what it has to do? It has to become hypotonic. So how to become hypotonic? The salt, all the salts are absorbed by the blood of vasa recta will be released from the ascending limb, and when salt is released, the osmolarity will increase drastically. So this is how counter current works. Counter current works inside the filtrate of Henle's loop. Counter current works inside the blood of vasa recta. One is responsible to creating this osmolarity, hyperosmolarity, and one is responsible for maintaining the gradient inside the kidney. Who is maintaining the gradient? Vasa recta. Who is creating the gradient? Henle's loop. So that was all about the counter current. Now you will ask me, sir, then what is the role of urea? What is the role of urea? You said urea is also responsible for maintaining the hypertonic medullary concentration. So you know that urea is reabsorbed through the distal part of collecting duct, right? So collecting duct releases urea here, and this urea will be accumulated into this zone in between the ascending limb and collecting duct into the inner medulla, and this accumulated urea will be taken by the thin segment of ascending limb, and continuously there will be recycling of urea in between ascending limb and collecting duct. Though majority of the urea will be released with urine, suppose 80% of the urea is released through urine, but 20% urea will be recycling into this loop. And while this urea is getting recycled, that is called urea recycling, that is not urea cycle. Urea is getting recycled. It is not urea cycle, urea is getting recycled. in between collecting duct and this ascending limb of Henle's loop, it will create a traffic jam kind of thing into the inner medullary zone, and that also contribute along with the salt particles to increase the hypertonicity of inner medulla. So that's why we said that salt and urea both are responsible for the hyperosmolar renal medullary zone. Now, next topic is regulation of kidney functions. Again, what actually kidney doing? There is blood flow inside the kidney. Kidney is trapping the blood to create a pressure for temporary basis, and then filtering out the unwanted material. So that's what kidney does, and it also helps in the process of osmoregulation of our body, maintaining water and electrolyte balance. Kidney also secretes some important hormones like erythropoietin, help in RBC formation. Kidney also secretes some other components, other factors like renin, which are responsible for maintaining the blood pressure BP of our body. So that's why you need to know the regulation of kidney function. The kidney and the heart they're related. Cardiovascular system and our excretory system, it's related to each other. So that's why when a kidney patient will be visiting a doctor, or a heart patient will be visiting a doctor, then doctor will also be checking for the kidney status. Okay. So it's written here, efficiently monitored and regulated by hormonal feedback mechanism involving the hypothalamus, JGA, juxtaglomerular apparatus, and a certain extent the heart. So this is how the regulation, kidney regulation works inside our body. So first it said that hormonal feedback mechanism involving the hypothalamus. For that we have to discuss the function of ADH, regulation by ADH, regulation by ADH. Okay. Because what is the source of ADH? Supraoptic nucleus of hypothalamus, and hypothalamus will be sending the produced ADH into the posterior pituitary, right? And when we require it, posterior pituitary will be releasing the ADH hormones or vasopressin hormone. What is the full name of ADH? Antidiuretic hormone. Now, what do you mean by diuresis? What do you mean by diuresis? Diuresis means excess loss of water through urination. Excess lot of water through urination. When your body is urinating hypotonic urine, that is called diuresis. And the opposite term is called natriuresis. Opposite terminology is called natriuresis. If your body is really means excreting out excess amount of salt, then that process will be called as natriuresis. So diuresis and natriuresis, they're kind of vice versa kind of phenomena. Now we'll be talking about the regulation of our kidney activity by ADH hormone. First, I want to tell you that the source of ADH is produced by hypothalamus. Hypothalamus produces the ADH by supraoptic nucleus, and then ADH is stored into posterior pituitary, the neurohypophysis. And when your body requires it, posterior pituitary will be, there will be secretion of ADH from this posterior pituitary. Okay. Now, at what condition? Suppose your body fluid is gaining saline, because if your kidney is losing water, your kidney is doing diuresis, means losing water So the salt concentration will increase into your blood, increase salt into our blood, so this is the excess loss of body fluid that will become blood saline, salinity will be increased, and that will be sensed by osmoreceptor that is present into your hypothalamus. That will send signal, the osmoreceptors of your body will send signal to your hypothalamus, the master of master, and upon stimulation of supraoptic nucleus, this special neural center present in your hypothalamus, supraoptic nucleus, that will release the hormone vasopressin or antidiuretic hormone. Now this ADH will go directly to the collecting duct. It will send signal to the collecting duct of your nephron, and the collecting duct will start producing or start activating special proteins to reabsorb water, aquaporin AP2, aquaporin 2, water, reabsorbing water, then there will be reabsorption of water. And while this like water is reabsorbed, where they will go? Reabsorption of water through the blood, the excess water will get inside the blood, and volume of blood is increased. Volume of blood is increased by reabsorbing the more or more water, or you can say not increase, the volume of water will be maintained, and if volume of water is maintained, the GFR will be restored, or the GFR will also be increased. So that's how antidiuretic hormone works. Function of ADH. Okay. Another name of ADH is vasopressin. Another name of ADH is called vasopressin. It's nothing but ADH only, antidiuretic hormone only. So what do you mean by the vasopressin? Vaso means blood vessels. Pressin, the meaning of pressin, to press the blood vessel, responsible for constriction. So that means this hormone is responsible for vasoconstriction. This hormone is responsible for vasoconstriction, right? And if vasoconstriction is done, BP, blood pressure, will be increased, and if BP is increased, GFR will be increased. Now you said that's how it can be possible. Can you provide some elaboration on this matter? Yes, the vasoconstriction of vasoconstriction of efferent arteriole will be there. vasoconstriction of efferent arteriole will be there. So it's like that. This is afferent arteriole and this efferent arteriole. More blood is coming, less blood is going. Now, if there is a vasodilation into your body due to the low blood pressure, so what will happen? So, less blood pressure will be produced into this glomerular wall and the GFR will decrease here. But vasopressin works upon this muscle of efferent arteriole. So, if there is vasoconstriction of efferent arteriole, if there is vasoconstriction of efferent arteriole, if there is low blood pressure, also then what will happen? If less blood is coming, the almost blood will not get any passage through the efferent arteriole, or very less blood will go out or get passage through the efferent arteriole. So ultimately what will happen? If blood is entering through the afferent arteriole and if you are pressing the efferent arteriole, then what will happen? The excess blood will be accumulated into glomerular region and ultimately that will increase the blood pressure and increase the GFR. So that's how the vasopressin hormone works here. Okay. Now, regulation of kidney by RAS, renin-angiotensin aldosterone system. That is called a RAS mechanism. Now again, when your RAS will be activated, if there is low BP into your body, there will be low GFR. Then your body will try to increase the BP as well as GFR, then your body will be activating the RAS mechanism. Yes, the low BP is sensed who will be sensing the low BP? Baroreceptor JG cells will be sensing the low BP. JG cells will be sensing the low BP. And if there is low GFR, the osmolarity will be sensed by macula densa cells. If there is low GFR, the fluid is flowing very slowly. If there is low GFR, the fluid is flowing very slowly into your renal tubule. So there will be more reabsorption of water and salt through PCT. So what will happen? The filtrate ultimately reaching to the DCT will become hypotonic, more hypotonic, and that will be sensed by the macula densa cell present on the wall of DCT. And once macula densa cell is feeling there is change in osmolarity, they will give signal to the JG cell, and this JG cells will be releasing, macula densa cell will be giving signal to JG cell, and JG cell will be releasing renin. Now renin will be mixed, will be released into blood, and inside your blood you have another component, that is angiotensinogen, released by your liver. Your liver produces this angiotensinogen. The renin inside the blood that will act on angiotensinogen, and this angiotensinogen will be converted into angiotensin 1. Now the same angiotensin 1 with your blood circulation will go towards your lungs, and the alveolar capillaries of your lungs will be releasing particular enzyme called angiotensin converting enzyme, ACE, angiotensin converting enzyme. So that will convert this angiotensin 1 into angiotensin 2. Now this angiotensin 2 is a strong vasoconstrictor. Now this angiotensin 2 will go to your adrenal gland, adrenal cortex. It will be stimulating the adrenal cortex region, and your adrenal cortex will release aldosterone. This aldosterone will come to the DCT and will stimulate the conditional reabsorption of salt and water. And if the water is absorbed into your blood, the volume is restored. If the volume is restored, the BP is restored. If BP is restored, GFR is also restored. Again, the angiotensin 2 is a vasoconstrictor. It will act upon heart. It can increase the cardiac output. It can the vasoconstrictor that has effect vasoconstriction of efferent arteriole. Vasoconstriction of efferent arteriole. If efferent arteriole is constricted, blood pressure inside the glomerulus will increase and the GFR will be increased. That's how this RAS system works for your body. Okay. So in this detailed diagram, the RAS is explained. The same thing is written here. If there is low BP into your body, there will be low GFR. And if there is low GFR, that will produce hypotonic filtrate. And this hypotonic filtrate will be sensed by your macula densa cell of juxtaglomerular apparatus. This macula densa cell, which are acting as osmoreceptor, they have sodium sensor. So if hypotonic filtrate means less sodium is there, macula densa cell will stimulate Lassis cell and Lassis cell will stimulate JG cell, which are also acting as a baroreceptor. Basically, JG cells are nothing but modified smooth muscles, and this JG cell will be releasing renin, hormone cum enzyme. The renin will get mixed into the bloodstream and it will cleave an enzyme called or proenzyme produced by liver called angiotensinogen, which is made up of 485 amino acid. Now this renin will cleave this angiotensinogen into a 10 amino acid long polypeptide chain called angiotensin 1. This angiotensin 1 is a mild vasoconstrictor. It has the capability to constrict the blood vessel to increase the blood pressure. But that mild vasoconstriction is not that much efficient. So with the blood circulation, it will reach to the lungs, and when it's passing by the alveolar capillaries, the alveolar capillary will release angiotensin converting enzyme, and that will cleave the angiotensin 1 into smaller polypeptide chain, 8 amino acid long polypeptide chains called angiotensin 2, which is a potent vasoconstrictor. This angiotensin 2 acting as a strong vasoconstrictor. The receptors of angiotensin 2 are called 81 and 82. They are present majorly on the efferent arterioles. Okay. So this 81 receptor present on the efferent arterioles will be stimulated by angiotensin 2, and that will cause vasoconstriction of efferent arteriole, increase in BP of efferent inside the glomerulus, and that will set the GFR or increase the GFR. Angiotensin 2, the receptors are present into our brain as well. They will stimulate the thirst receptor of hypothalamus. So what will happen? Thirst receptors will tell you you are thirsty, drink a lot of water, you will start drinking. It will also increase the salt appetite. If you are taking more salt, you will become thirsty, and the next movement, you'll start drinking more water. So it will increase your water drinking capability and salt appetite, so that your blood volume, if you are drinking more water, more fluid intake, blood volume will be increased and GFR will be increased. Okay. Angiotensin 2 also act on the brain to release more ADH. It will tell hypothalamus secrete more ADH. It will stimulate sympathetic nervous system. It will stimulate sympathetic nervous system to increase the heartbeat and increase the reabsorption of water through nephron. Most importantly, it will stimulate zona glomerulosa of adrenal cortex. Adrenal cortex has three layers: zona glomerulosa, zona fasciculata, zona reticularis. So it will stimulate particularly ZZ, zona glomerulosa of adrenal cortex, and this zona glomerulosa will release aldosterone hormone, right? Aldosterone hormone, uh, corticosteroid, one of the corticosteroid hormone, and that will act upon mainly on DCT and to some extent on collecting duct, and that is responsible for conditional reabsorption of mainly sodium ion and water, also responsible for secretion of potassium and H plus ion. So aldosterone is not only responsible for absorption of salt and water, is also responsible for secretion of selective secretion of potassium ion and proton or H plus ion, and that's how it's helping us to retain body water. So that's how RAS is helping us to increase the blood pressure, increase the maintaining the GFR. How our kidney will be deciding either to increase the blood pressure or decrease the blood pressure, and obviously blood pressure is increased, GFR will be increasing, blood pressure is decreased, GFR will be decreasing. Now we need to increase the blood pressure where? We need to increase the blood pressure into the glomerulus. Yes, as because the due to the pressure created into the glomerulus endothelial wall, there will be filtration and gradually initiation of formation of urine, right? So let's check again the diagram of the Malpighian corpuscles. This is the Bowman's capsule. Along with glomerulus, we call it Malpighian corpuscles. This is the afferent arteriole, glomerulus, and efferent arteriole. Afferent arteriole is always wider than efferent arteriole. You need to remember this. AA for afferent arteriole, EA for efferent arteriole, G for glomerulus, B for Bowman's capsule. Okay. Now let's check the direction of blood flow. Blood is entering through afferent arteriole and exiting through efferent arteriole. Due to the variation or difference in their width, there will be accumulation of extra or excess blood, and that will be creating pressure on the wall of glomerulus. Okay. Now, when or how we can increase the BP here inside the glomerulus or decrease the BP inside the glomerulus? Now, to increase or decrease the blood pressure, we are acquainted with these terms called vasoconstriction and vasodilation. Vasoconstriction and vasodilation. Okay. Now we were saying that angiotensin 2 is a potent or strong vasoconstrictor. Vasopressin, ADH, is a strong vasoconstrictor, and due to that vasoconstriction, blood pressure increases, GFR increases. So when we're talking about vasoconstriction, we need to constrict the efferent arteriole. If the lumen of efferent arteriole is constricted, if the lumen of efferent arteriole is constricted, the channel will be narrowed down. The channel will be narrowed down, and if channel is narrowed down, automatically less blood will be flowing outwards and more blood will be accumulating in the glomerular region to increase the blood pressure here. If blood pressure increases, obviously glomerular filtration rate, GFR, gonna be increased. So that's how vasoconstriction works. Now, if you ask that if we, instead of constricting the efferent arteriole, if we perform vasoconstriction of afferent arteriole, then what will happen? If you constrict the afferent arteriole, then less blood will be entering through it, and if less blood is entering through afferent arteriole, accumulation of less volume of blood will be there inside glomerulus, and that will be decreasing BP, decreasing blood pressure here. So it's very tactful or very tricky to understand when you're talking about vasoconstriction or vasodilation. Will it be constricting afferent arteriole or afferent arteriole? Similar things goes for vasodilation. Okay. Now, if you vasodilate efferent arteriole, then what will happen? More blood is coming through afferent arteriole and more blood is going out through efferent arteriole. So less volume of blood gonna stay here into glomerular region. So there won't be any improvement in blood pressure. So if you vasodilate efferent arteriole, your BP will be decreasing. Similarly, if you vasodilate your afferent arteriole, if you vasodilate your afferent arteriole, you are allowing more volume of blood to enter, and that will be increasing blood pressure. For vasoconstriction, if you are vasoconstricting efferent arteriole, BP will increase. If you vasoconstrict afferent arteriole, BP will be decreased. So that is the conceptual understanding of vasoconstriction and vasodilation and how they are related to increasing or decreasing the blood pressure. And obviously GFR is directly proportional to the increase or decreased blood pressure. Let's move forward. Now we'll be talking about the regulation of kidney functions. Yes. Okay, already we have completed the regulation of kidney function of with RS with ADH. Now we'll be jumping towards the antagonist. Now, what do you mean by antagonist? Antagonist means is opposing. The RAS mechanism. Now we have here ANF, atrial natriuretic factor. Natriuresis, opposite phenomena for diuresis, right? Okay. Sorry, natriuresis means absorption of or release of salt particles. Now this ANF, atrial natriuretic factor, it will be promoting natriuresis. Now what is natriuresis? Natriuresis means excretion of salts, particularly sodium and water with kidney. So natriuresis means excretion of salt or minerals and water through kidney. It's called natriuresis. So it's trying to check, it's trying to make an adverse effect of RAS as well as the mechanism initiated by ADH, right? So what will do? The atria of heart will secrete ANF, atrial natriuretic factor. Even few others organism in our body can produce this kind of natriuretic factor. Brain will be producing BNF, brain natriuretic factor. There are different kinds of natriuretic factors, and majorly ANF is considered into our NCERT. So this atrial natriuretic factor will try to inhibit RAS and ADH activities, And if RAS and ADH activities are inhibited, so it will promote natriuresis. It will promote or give positive signal for natriuresis, excretion of sodium chloride and water through kidney. And what will happen if there is exaggerated BP in your body, exaggerated or increased BP in your body, that will stimulate secretion of arterial natriuretic factor from atria of heart, and ultimately that will be decreasing the blood pressure, and if BP is decreased, GFR, glomerular filtration rate, will be decreased and the aim is achieved. Let's move forward. Now the topic is micturition. Yeah. Now what is micturition? Micturition means the process of uh, eliminating urine which is stored into the urinary bladder, right, by creating a reflex called micturition reflex. Yeah. So this is the process of release of urine. Micturition is the process of release of urine. Now, how it is achieved? Basically, it's a neural mechanism causing, it is called the micturition reflex. So, the neural mechanism which is responsible for causing the micturition, that is called micturition reflex, mainly which is initiated by our CNS, central nervous system. Okay. Now, some data is given here. An adult human excrete on an average dash liters of urine per day, 1 to 1.5 liters of urine per day, right? The volume can vary person to person depending upon the salt intake or water intake or fluid intake. The urine formed is a light yellow colored watery fluid. Now, why it's light yellowish color? Because it has the remainings of the bilirubin from the liver called as urobilin. Urobilin, which is slightly acidic in nature. What is the pH of urine? It's written here. pH is 6 and has a characteristics order due to presence of some VOC, volatile organic components, and we call them urinodes, right? Okay. Now, while we are passing urine with urine, we are mainly excreting urea because we are ureotelic. So, on an average, 20 to 30 grams of urea is excreted out per day. You need to remember these informations. Directly statement based question can be asked. The volume of urine released per day and the amount of urea is excreted out per day for a healthy human being. Now, why we need to study all these things? What is the importance behind this? The importance is written here. So, various conditions. Why do we need to calculate the volume of urine we are excreting? Why do we need to calculate the amount of urea we are excreting? So, the various conditions can affect the characteristics of urine by observing the color, by observing the odor, or by pathological test of the urine presence of excess amount of glucose, excess amount of ketone bodies, excess amount of urea, excess amount of creatinine. We can diagnose different kinds of conditions or disorders or disease inside our body. Okay. So, analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. That is very much important. Ultimately, Kidney is one of the most important organ of our body. It's related to our circulatory system as well. If your kidney is not functioning properly, that will affect your heart, this will affect your cardiovascular system. For example, presence of glucose. That condition is called glycosuria. Glycosuria, glucose in urine. Glucose in urine is called glycosuria. And ketone bodies, and the condition is called ketonuria. If there is diabetes or hyperglycemia in our body, then that gonna release that excess glucose into your urine. Though glucose is a high threshold compound, in normal condition, if your body is facing hyperglycemic condition, it will not be excreting out glucose even through the process of urination. So if there is a if glucose crosses the limit, if it crosses the threshold level, threshold limit, and then only you will be getting glucose inside your urine, and that is like that presence of glucose inside your urine, that's a significant rise in glucose in your body, and obviously that's denoting towards diabetes mellitus, means when urine is becoming sweet. Ketone bodies, how ketone bodies are formed? Even there can be presence of ketone bodies after prolonged fasting. But again, if you are suffering from diabetes mellitus, there will be presence of excess ketone bodies, and they are acidic in nature, and that will be calling ketoacidosis. Your urine will become much more acidic in nature. The pH will fall drastically below 6. Yes. So, due to the fat metabolism, fat catabolism, these ketone bodies are produced, and the condition is called ketonuria in urine, are indicative of diabetes mellitus. So there are two conditions. One is diabetes mellitus, one is diabetes mellitus, and another is diabetes insipidus. Diabetes mellitus and diabetes insipidus. Now, what is the difference between them? Diabetes mellitus means sweet urine. Though we are not gonna taste urine, but that's because there is glucose inside that urine, it may taste sweet. So that's that condition. The name is derived from this. Now, diabetes insipidus. Insipidus means tasteless urine, colorless or tasteless urine. Colorless and tasteless urine is because there is excess secretion of water with your urine, and when you will be suffering from diabetes insipidus, when there is low or hyposecretion of ADH into your body, then you will be suffering from diabetes insipidus, and when there is hyposecretion of insulin, then you will be suffering from diabetes mellitus. And obviously, if you are suffering from diabetes mellitus, there will be glycosuria, glucose in urine, there will be ketonuria, there will be ketoacidosis. So that's all it's written here. If there is more ketone bodies present into your urine, if there is ketonuria, there will be obviously ketoacidosis, because these ketone bodies are organic acids mainly. So that's why urine has very importance. The like pathological diagnosis of urine, the presence of the components has importance in detecting the metabolic disorders into our body. Okay. Now let's come into the process of micturition. So we can say that pathological diagnosis of urine and the components present into it is very much important to denote the body condition as well as the kidney health. Now let's come to the topic, that is micturition, and as per the NCERT, we'll try to learn the process. Okay. So basically, inside the urinary bladder, this is the urinary bladder, there will be accumulation of urine gradually. When the urinary bladder is 1/3 filled, when the total volume of the urinary bladder of a healthy mature human male is nearby 800 to 900 ml. When it's 1/3 filled, like near about 300 to 400 ml urine is already accumulated inside the urinary bladder, that is going to create a tension that will increase the tension inside that urinary bladder. It has the transitional epithelium, right? So it will cause stretching of the receptors present into the detrusor muscle, the muscular layer present into the lining of this urinary bladder. So basically thing is like that, urinary bladder is filled 1/3, then you can really sense your urinary bladder is getting filled with urine, the urge of urination you can really feel. So that is due to the increased tension into urinary bladder, and when the tension is increasing in the wall of UB, then it will stimulate the stretch receptors present, and this stretch receptor will give signal to your central nervous system. Okay, now basically what kind of signal it's sending to CNS? It's triggering the micturition reflex. It's triggering the micturition reflex. So with triggering the micturition reflex, the stretch receptor on the wall of this urinary bladder. Okay. Now CNS is giving a positive motor signal as a component of the micturition reflex, and the motor signal will be signaling to the sphincter. The opening of urinary bladder, or you can say urethra more precisely, that is guarded by two sphincters, one internal involuntary sphincter and one external voluntary sphincter. So basically, it will be stimulating the external sphincter, which is a kind of voluntary phenomena, and there will be micturition. You will start releasing out or eliminating out the urine present inside that urinary bladder. So that was a very simplified explanation of the process of micturition. Again, I am repeating it. Kidneys are filtrating your blood, and that formed urine inside the kidneys are coming through these ureters, and gradually these urines are getting deposited inside the urinary bladder. When the urinary bladder is 1/3 filled, that force or the pressure exerted by the urine is enough to create a panic in the muscles of this urinary bladder. We call them detrusor muscle. So that's why it said that the tension is increasing gradually. When the tension is increasing into the urinary bladder, that will give us positive signal to the stretch receptors. When the stretch receptors are stimulated, they can feel, the urinary bladder can feel that yes, my volume is increasing, I am getting stretched. So that will directly give a positive signal to the central nervous system, and that is called the micturition reflex is triggered. The micturition reflex is triggered. Now as soon as the central nervous system can detect this trigger, this signal, it will send another motor signal, positive motor signal, to the voluntary sphincter present on the out or exit opening of the urethra related to this urinary bladder, and that will cause opening of external sphincter, and if sphincter is opened, it will cause micturition voluntarily, right? So basically we can hold the pressure of urination for a certain time being due to, again, the voluntary or the sympathetic actions of our nervous system. And when basically you are releasing the urine, that sympathetic action will be inhibited for a time being, and you can urinate. And even if you want, and the second means the second for a second or for 2 seconds, you can again inhibit the motor signal, and you can close the sphincter and you can stop your urination for a time being. So that's why it said that it's voluntary mechanism. Okay. Now we'll be learning in detail about the micturition in detail. So basically, we'll be discussing micturition. It's regulated by central nervous system. It has voluntary regulation. It has involuntary regulations as well. Now, involuntary regulation means autonomic nervous system. CNS has autonomic nervous system, and CNS has somatic nervous system. And the autonomic nervous system has the parasympathetic part. Autonomic nervous system has sympathetic part. Both of them are antagonist to each other, parasympathetic and sympathetic. Now let's discuss about the function of the parasympathetic part of ANS, how it's affecting your urinary system. So parasympathetic nerve is called nerve of filling because it will be stimulating the process of filling of urine inside your urinary bladder. So parasympathetic nerve that will be dilating, is responsible for dilation of urinary bladder. Basically, it will be relaxing the muscles of urinary bladder. We call them detrusor muscles. So ANS is divided into parasympathetic nervous system and sympathetic nervous system. ANS has the parasympathetic part and sympathetic part. Sympathetic is called as nerve of filling. Parasympathetic is called as nerve of emptying the urinary bladder. Why and how? So basically, sympathetic nerves, we call them nerve of filling. How they're performing so? They're responsible for relaxation or dilation of urinary bladder muscles. That is called detrusor muscle, and as well as they are performing constriction of internal sphincter. So this is how they are performing. So if this is the normal shape of your urinary bladder, and if this is the internal sphincter, upon the stimulation of sympathetic nerve, the volume of the urinary bladder will be increasing. The volume of urinary bladder will be increasing due to the dilation or the relaxation of detrusor muscle present into the wall of urinary bladder. Similarly, it is responsible for closing the internal sphincter, that is called constriction of internal sphincter muscle. So if internal sphincter is closed and the urinary bladder is getting dilated, so that will cause filling of urine and storage of urine inside the urinary bladder. But in case of parasympathetic nervous system, parasympathetic nerves are called nerves of micturition or nerves of emptying the urinary bladder. So how it will be emptying the urinary bladder? It will cause the constriction of the muscles present into the urinary bladder, or constriction of urinary bladder muscles. So the volume of urinary bladder will be decreased. If this is the initial volume of urinary bladder, the volume will be decreased upon action of parasympathetic nerve. The volume is getting decreased now. And secondly, it will inhibit the sympathetic nerve, so there will be relaxation or dilation of internal sphincter. So when urine was getting filled into the urinary bladder was relaxed and internal sphincter was constricted and closed. Upon stimulation of parasympathetic nerve, urinary bladder will decrease the volume by performing constriction. The detrusor muscle into the urinary bladder will get constricted, and that will squeeze, create pressure of the urine to release or get eliminate out from it. And how to eliminate? You need to open the internal sphincter. So the passage of internal sphincter is opened now. So easily you can micturate the urine outwards. But there is a problem right now. So what's the problem? The exit pathway, you got another sphincter that is closed. That is the external sphincter. So you have two sphincter, internal sphincter and external sphincter. Now we are done with the functioning of the internal sphincter. Internal sphincter in normal condition, you'll find it into closed condition when it's stimulated by sympathetic nervous system. When the urinary bladder is filled, then which will be predominant? Parasympathetic nervous system will become dominant now, and that will open by relaxing the muscle of internal sphincter, and the urine is coming out, and that is creating pressure on your external sphincter. Now really you are in a urge to pass the urine from your excretory system, but as because you are an adult person, anywhere you cannot. Suppose you are sitting in a class, you are sitting in an office, you cannot pass the urine, right? Now you need to find a urinal or washroom, and there you can go and you will start urinating. Till then you need to hold the external sphincter. Till then you need to hold the external sphincter. Now how to hold? That is depending upon the action of somatic nervous system. So our somatic nervous system in normal condition is giving positive signal to the external sphincter to the external sphincter and your external sphincter is closed, or you can hold the pressure of urination. But once the micturition reflex is initiated, once the micturition reflex is initiated, that will inhibit your somatic nervous system by giving negative signal, and that will give signal to your external sphincter. Now external sphincter will be open, and when your external sphincter is open, you can micturate. So that's how the complex mechanism of micturition works. Again, I am simplifying it. So in normal condition, when you are not micturating, you are, suppose you are sitting in a class, you are listening to your teacher. Now in that time, your body is performing the filtration of blood inside your kidneys. Your kidneys are filtering the blood, forming urine and sending into the urinary bladder with the help of ureters. So that is called the filling of your urinary bladder, and in that time your urinary bladder is who is dominating your urinary bladder? The sympathetic nervous system is dominating your urinary bladder, because it's just the filling of your UB, urinary bladder. So at that time, the muscles of your urinary bladder is relaxed, and the internal sphincter is closed upon action of sympathetic nervous system. But gradually your urinary bladder is getting filled, creating pressure. Now then sympathetic nervous system will gradually is becoming switched off, and parasympathetic nervous system will switch on. Now it will become the dominant one. Now the parasympathetic nervous system will send signal to your urinary bladder to become constrict, to squeeze out the urine, right? So that is due to the constriction of urinary bladder muscles. Primarily we call them detrusor muscles. And due to the activity of parasympathetic nerves, the internal sphincter will be opened now. Now gradually the urines are coming out from urinary bladder. But before urination, the urine needs to cross another barrier, that is called the external sphincter. But parasympathetic nerve cannot open the external sphincter, and thank God we have this external sphincter. That's why we can hold the pressure of urination. So you are holding the pressure of urination due to the activities of somatic nerves. So how you can hold the pressure of urination? Somatic nerves always give positive signal to the muscles of the external sphincter, and that's why external sphincter remain closed normally. But now you need to urinate. You are in a urinal, you are inside the washroom. So in that case, when the micturition reflex will be generated into your body, that will inhibit the somatic nerves controlling the external sphincter. And when the somatic nerve function is inhibited, the external sphincter will be opened for a time being, and you will be micturating, you will be passing the urine. So that was the whole science behind the process of micturition. Now let's move forward to the roles of other organs in excretion. So we know that we have one pair of kidneys for the process of excretion, but these organs also helping us every time to excrete some of the unwanted materials inside our body. So first we'll be talking about lungs. We know that lungs, they are the respiratory organs, biological machines for respiration present into our body, into our thoracic cavity, surrounded by the rib cage, right? So lungs, basically inside lungs, what's happening? Exchange of gases happening inside the lungs. So the carbon dioxide produced during the process of intracellular respiration, that's a major excreta produced inside our body. The excess amount of water vapor produced during the time of respiration, intracellular respiration, that's also a kind of excreta for us. So basically lungs are responsible for excretion of carbon dioxide. Now how much carbon dioxide is excreted out? 200 ml per minute. Carbon dioxide, 200 ml of carbon dioxide is excreted out per minute while we are respiring. And water, how much water we are excreting out? Near about 400 ml water per day, nearly 1 cup of water. Okay, so that's how our lungs are contributing in the process of excretion. Let's come to the function of liver. Yes, liver is called detoxifier of our body. So liver is responsible for excretion of drugs, whatever we are taking, like in the form of medicines, vitamins, excretion of drugs, vitamins, cholesterol, bile pigments. Now, what do you mean by bile pigments? Bilirubin, biliverdin, they are called bile pigments. So these are the major excreta excreted by liver: drugs, vitamins. Okay, then also degraded steroid hormones. So that's why we call liver as detoxifier body. This is the largest gland of our body as well. Next, saliva. The secretion of salivary glands are called saliva, and with saliva we release water. 99.5% of saliva is water only. Now that is mixed with sodium chloride, Na plus and Cl minus, sodium ion and chloride ion. With saliva, we also release a little amount of uric, nitrogenous wastes are also released or excreted with saliva. There is a little amount of urea. Little amount of urea is also excreted with saliva. Now, sweat glands and sebaceous gland, they are present into our skin. Yes, with the help of our skin also, we are excreting out some of the unwanted materials produced inside our body. Now, sweat glands are also called as sudoriferous gland. The sweat is also called as sudor. So, how they are helping us? They are helping us in the excretion of excess water, excess salt, lactic acid. This sort of things are getting eliminated out through our body, right? And a little amount of glucose and fatty acids are also secreted or excreted with the help of our sweat glands. You need to remember this, you need to know this, that we do not excrete any of the nitrogenous waste through our sweat, but we excrete nitrogenous waste through our saliva. Now, sebaceous glands, they are also present into our skin. They are called as oil glands. Okay, one more thing about sweat gland. The question can be asked, what is the primary function of sweat gland? Excretion is the secondary function. What is the primary function of sweat gland? It's responsible for maintaining the body temperature. So cooling effect. Cooling effect is the primary function for the sweat glands, and how it's doing so. When the sweat is getting evaporated, that's collecting latent heat from our body, and that gives us cooling effect. Sebaceous gland, that secretes the sebum, the oil. Oil is called the sebum. Now, why our skin is releasing or secreting oil? That will produce a protective lining, protective film over our epidermis, and that will protect against uh, different kinds of microorganisms that will help us, that is acting as a moisturizer for our skin. And with the help of the sebum secretion, we are excreting out wax, sterols, fatty acids, etc. Now, sweat gland and sebaceous gland, they are present inside our skin. Yes, our skin is also helping us to excrete some of the unwanted products, right? So, when you're talking about sweat gland, they're also called as sudoriferous glands. Sudor is another name of sweat. Yes. Now, what is the primary function of sweat gland? That is providing the cooling effect of our body while a sweat on your skin. When sweat on your skin is getting evaporated, that is collecting latent heat from your body, and that's only giving you the cooling effect. So that is the primary function. The primary function of your sweat gland is to give you cooling effect. Okay, now what are the substances getting excreted out with your sweat? Now your sweat contains water, salt, lactic acid, then a little amount of urea, glucose, as well as fatty acids. So all these components are excreted out with the help of your sweat gland. Now let's come to the sebaceous gland. Sebaceous glands are also called as oil glands on skin. Their oily secretion is called sebum, and they are forming protective film on your epidermis, forming protective film or protective covering of your outer layers of your skin. And with the help of this sebum, you are excreting out sterols, wax, fatty acids, and other hydrocarbons. Now, we have come to the last topic of the chapter, that is disorders of excretory system. And the disorders are uremia, glomerulonephritis, renal calculi, renal failure. First, what do you mean by uremia? Uremia means high urea concentration into our blood. That's because your kidneys are not working properly. So if you're facing consecutive uremia for a prolonged time duration, chronic uremia for a prolonged time duration, that is the indicative of kidney failure. So high concentration of urea in your blood is because the urea is unable to getting filtered out with the help of your nephrons. Okay. Now, if you are diagnosed with uremia, the next test you have to perform, that is either your glomeruli are functioning properly or not. Then possibilities are there. You may face inflammation of your glomerulus or glomeruli in plural, as because they are getting damaged, right? And that condition is called glomerulonephritis. Glomerulonephritis, it's ending with itis, and that is the meaning of inflammation. If you're getting a term and the term is ending with itis, glomerulonephritis, conjunctivitis. So the itis means inflammation. So inflammation of your glomerulus is because they are getting damaged due to any infection or due to the autoimmune attack of your body. And obviously, if your glomerulus or glomeruli are unable to work properly, they're not gonna filter your blood in a proper manner, and that gonna give rise uremia. And next disorder here you got, that is renal calculi or kidney stone. Renal, this word is related to kidney. Now renal calculi means kidney stone. Calculi, the word came from calcium, deposition of calcium salts into your kidney. Basically, these are insoluble crystalline stones, and majorly it's calcium oxalate is responsible for kidney stone. Now, next, a very fatal condition, that is renal failure or kidney failure. Now, this kidney failure, there are many solutions. One is temporary solution and one is permanent solution to treat kidney failure. Now, what is the temporary solution? You have to go for dialysis. The frequency may be within every week or every 4 days or every 2 days, depending upon the situation, the accumulation of nitrogenous waste in patient's body. But if you want a long-term solution or permanent solution, you need to go for kidney transplantation. Kidney transplantation, and for kidney transplantation, donor is needed. Okay. Now, in case of kidney transplantation, doctor will ask that if you are getting a donor, he or she must be one of your close relative with similar blood group, with similar blood group and similar HLA. There should be tissue match. If blood group and HLA, human leukocyte antigen, is not matching with your body, if you are the patient, and with the donor, then your immune system, basically your T cells, gonna reject the implanted or the transplanted kidney into your body. Okay, now one more thing here. If you are going for kidney transplantation, you don't have to remove the failed kidneys, right? Because kidneys are not only responsible for excreting out or eliminating out nitrogenous waste, your kidneys are doing so much more for you. So that's why when a patient is performing a doctor is advising a patient to perform kidney transplantation, the doctor will not be taking out the failed kidneys, but instead, inside the peritoneal or the retroperitoneal region of that patient, doctor will install another healthy kidney. So now the person or the patient has 3 kidneys inside the body. Remember this. So let's elaborate a bit about the process of hemodialysis. Hemodialysis means the blood of the patient, is accumulating nitrogenous waste. That's because kidney is failed, kidney is not working properly. So you need help of artificial kidney. We need help of artificial kidney to substitute the function of the failed kidneys inside our body. And the artificial kidney, basically it's a machine that's called hemodialyzer. The machine is called hemodialyzer. Now, how it will be performed? Hemodialysis. You need to create channel inside the body of a patient, and by choosing the proper artery and creating the channel into the artery, you need to draw out the arterial blood, as because inside the artery the force of blood or the velocity of blood is quite higher. So that's how we are channeling out the blood, the systemic blood from the patient's body, which is rich in nitrogenous waste. Now, as because we are drawing out from the patient's body, you need to you need to mix it with anticoagulating agent like heparin or other anticoagulating or other synthetic anticoagulating agent, and then we are putting this blood inside the dialyzer machine. Now these tubes are used to draw the blood. They are made up of cellophane. Okay. Now inside the dialyzer, the blood is moving forward. The fluid inside this dialyzer that has the similar concentration of your plasma, that is near about 300 milliosmol, but that is lack with urea, ammonia and other nitrogenous waste. So there will be a creation of gradient in between your blood and the fluid present into the dialyzer machine. So what will happen now? The urea, ammonia or the nitrogenous waste will start moving towards the concentration gradient, right? And your blood is free of the nitrogenous waste. So the dialyzer machine worked as a kidney. It has removed the nitrogenous waste from your blood, and the waste free blood will be drawn out from the dialyzer machine, will be added with antiheparin to neutralize the excess amount of the anticoagulant you had added, and now we'll be channelizing the filtered blood into the vein. So that's how the process of hemodialysis or dialysis of blood is performed. Okay, here in detail machine of hemodialyzer is described now. Once you can download the PPT or the PDF of this particular lecture from our PW app, you can study in detail. Okay, now let's solve some of the questions. And the first question is given here, that pyramids, renal pyramids in a kidney of human are. So pyramids are equal in the number of minor calyx, and the answer will be 12, right? So the number is nearby 10 to 12 or 8 to 12. So 12 is our proper answer. Renal papilla is a part of inside your kidney. You have cortex and medulla. Medulla has medullary pyramids, right? Now, the conic part of medullary pyramids are called renal papilla. So they are part of renal pyramids. Next question, assertion and reason. The assertion is saying that nephrons are of two types, okay? They're cortical and juxtamedullary nephron based on their relative position in cortex and medulla. That is true. Reason is said that because juxtamedullary nephrons have short loop of Henle. Juxtamedullary nephrons have short loop of Henle. No, cortical nephrons have short loop of Henle. So that is a wrong statement, whereas cortical nephron have a longer loop of Henle. So that's a false statement. So we can say that simply assertion is true, but reason is false. Okay. Now a chart is given, flow chart. Select the correct order of osmolarity changes in filtrate with reference to plasma, Bowman's capsule, then PCT. Okay, so it will be like this: Bowman's capsule, then PCT, then descending limb, then ascending limb, then DCT, then collecting duct. Okay, so let's talk about that osmolarity. So we know that osmolarity of Bowman capsule, the filtrate inside Bowman's capsule is 300 milliosmol. Inside the PCT is isoosmolar to the filtrate within the Bowman's capsule, that is 300 milliosmol. Descending limb towards the U loop, it is gaining the osmolarity of 1200 milliosmol, getting hypertonic. And when we are following the ascending limb going towards the DCT, it's again becoming hypotonic. Inside the DCT, it's again becoming hypotonic, and when it's passing the collecting duct as urine, it's becoming hypertonic. Okay, so Bowman's capsule, it will be the isotonic with reference to our blood or plasma. The blood or plasma, the concentration of fluid is 300 milliosmol. So in Bowman's capsule is 300, that is isotonic. That's great. In PCT, that is also 300, that is also isotonic. In descending loop of Henle, that is hypotonic. No, that is hypertonic. So this option cannot be granted. Let's check the second option. Bowman's capsule is isotonic, 300. In PCT, 300, isotonic. Descending loop of Henle is 1200, hypertonic. In ascending, it's again becoming 1200 to 1800, 300 and more. So it will also become hypotonic again. In DCT, it's hypotonic. In CD, end of collecting duct, it's hypertonic with 1200 milliosmol. So we got our perfect answer. That is option 2. Have you got it? Why it's the answer? The concentration of blood is 300 or plasma is 300 milliosmol. The filtrate inside the Bowman's capsule that also has similar concentration, that is isotonic. Similar concentration in PCT, 300 milliosmol, that is isotonic. While entering into the Henle's loop, the descending arm, gradually it will become 1200 milliosmol, so it's hypertonic. When it's entering to the ascending loop, going upwards, is becoming hypotonic. Entering into the DCT, losing salt and water, becoming more hypotonic. And upon entering into the collecting duct, when it's released out from the collecting duct or the distal part of the collecting duct, it will gain 4 times more osmolarity than your body fluid, that is 1200 milliosmol, that is again hypertonic. That's why this is our correct answer. Okay, now come to the next question. If Henle's loop were absent from the mammalian nephron, if the nephron is like this, if there is no U-shaped loop, then what will happen? There will be no urine formation. Urine will be formed, so this can't be the answer. There will be hardly any change in the quality and quantity of the urine. That can't be the answer. The urine will be more concentrated. No, without Henle's loop, how urine can be more concentrated? The urine will be more dilute. Yes, this is the answer. Because we know about the counter current mechanism, who is responsible for counter current multiplier to increase the concentration of the osmolarity of the inner medullary zone, that is Henle's loop. Who is responsible for maintaining the counter current exchanger mechanism is performed by vasa recta. So if there is no Henle's loop or no vasa recta, there will be no counter current. So simply the urine you will be excreting out, that will be with a lot of water, there will be diuresis, and that condition gonna affect your body health. So that's called diluted urine. Okay, now another good question we got here. So here we need to study a graph. This is point O, this is point X. Okay, so as per the question, we can see here the graph shows the effect of liver and kidneys in dogs on the amount of urea in the blood. Okay, in the liver and kidney, and you have to find out the presence of urea in the blood. Okay, on the graph, 2 marks O and X shows the time when these 2 organs were removed. Okay, you need to remove liver and kidney. Now, which mark shows the removal of which organ. Now see, if you know the whole process, our body produces ammonia as a excreta. Now, as because the ammonia is very much toxic, our body will send it into the liver, and by the process of ornithine cycle or urea cycle, our liver will convert the ammonia into urea, right? Now, your urea will be mixed into your blood, and our body will fetch it into kidney, and with urine the urea will be excreted out. The urea will be excreted out, and similarly inside your kidney your blood is filtered out. There is no urea present inside the blood after crossing the filtration process inside your kidney. So if I remove liver from your body, then what will happen? If I remove kidney from your body, then the concentration of urea will drastically increase into your body. If I remove kidney, because kidney is responsible for the removal of urea from your blood and putting into urine, excreting it out. So if I remove kidney drastically, there will be uremia into your body, and the urea graph will go higher. This is the graph of urea, concentration of nitrogenous waste in blood. So upon removal of kidney, so you can say that O is kidney. Upon removal of kidney, the graph will go forming a slope going upwards. The concentration of nitrogenous waste and urea will be increasing, and you can say that X is the removal of liver. If liver is removed from your body, then what will happen? There will be only formation of ammonia, but there will be no formation of, no conversion of urea. So there will be no change in urea concentration into your blood. So our answer will be O stand for removal of kidney and X stand for removal of liver. Yes. Next question. Which of the following is responsible for excretion of dilute urine? Which of the following is responsible for excretion of dilute urine? More secretion of renin. Renin is responsible for initiation of RAS. That will increase the BP. If BP is increased, GFR will be increased. And if GFR will be increased, then there is the possibilities of secretion of or excretion of concentrated urine, but not diluted urine. Less secretion of vasopressin. Yes, vasopressin means ADH. If there is hypo secretion of ADH, then there will be diuresis. So that will lead to excretion of diluted urine. If there is more secretion of aldosterone, then concentrated urine will be produced. That is a part of RAS system. RAS is responsible. Renin, angiotensin, aldosterone, they are part of RAS. RAS is responsible for what? RAS is responsible when RAS is stimulated into your body. If RAS is responsible for increasing BP, increasing GFR, and that will help you to produce concentrated urine. Remember, less secretion of atrial natriuretic factor. If ANF is secreting less, then obviously there will be production of concentrated urine. If there is more secretion of atrial natriuretic factor, then there is possibility then your kidneys will be excreting out diluted urine. So this is also not our answer. This is also not our answer. This is also not our answer. We got the correct answer here. Okay. So this was all about the chapter. Now you can download the PDF of handwritten notes from PW app, and I have summarized the chapter into 3 pages. You can look over this, and we are ending this particular one shot. Thank you. Bye bye. Jai Hind.